Good morning. Welcome to the Danforth Center. I'm Jim Carrington, president of the Danforth Plant Science Center, and I'm thrilled to see so many people here this morning. I think we're going to have a lot of people rolling in as the morning gets off the ground. And it's a real honor to host this symposium on gene editing, innovation and impact in Missouri. And I really want to underscore that Missouri part because for those of you who are active in the ecosystem here and across the state, it's no surprise to you what's going on. For those of you who are a little less familiar, I think you're going to be surprised to see the scope, the breadth, the magnitude of what's going on here related to gene editing and innovation. If you walked around the Danforth Center today, you would find that gene and genome editing and a whole range of peripheral connected derived tools and technologies that have emerged from gene editing are ubiquitous and common in just about every laboratory here, as you would find at most medical schools, in many research institutions. These are both common and essential tools for discovery in modern biology. This morning, however, we're going to focus on how these tools and technologies are finding their way into the marketplace in human health and agriculture, and how these innovations are shaping our region today as well as in the future. We're excited to co-host this symposium with two longstanding partners of the Danforth Center, Benson Hill Biosystems and Missouri Cures. And I want to call out three individuals in particular: Natalie DiNicola, Chief Communications Officer for Benson Hill Biosystems. You'll hear from her later. Dina Ladd, Executive Director for Missouri Cures, and our own Carla Rober from the Danforth Center. Thank you, Natalie, Dina, and Carla. And thank you all for coming this morning. It's good to see you. Now, please join me in welcoming to the podium Dina Ladd.
Thank you, Dr. Carrington. I would also like to thank the Danforth Center and Benson Hill for co-hosting this event with us. The Missouri Cures Education Foundation is the organization in the state. We promote and protect medical research, and we host various research symposiums on different topics. And I recognize a lot of familiar faces in the audience, so thank you for your continued support of coming and learning about medical research. I would also like to thank today's sponsors for their support. They include the Steinberg Group, the Steinberg Foundation, Biotechnology Innovation Organization, Global STL, Goldenberg Heller Antonelli Law Firm, and Gamble Schlemeyer Governmental Consultants. And then I'd also like to acknowledge a couple of elected officials that we have with us. Senator Jill Shoup, thank you. And Representative Tracy McCreary. And I believe, so thank you for your support and for being here today. A few housekeeping items before we get started. We have three amazing panels for you this morning, and we will not be taking a break, but please feel free to get up and grab that second cup of coffee, or if you need to use the restrooms, they're located on this floor in the atrium. You were given cards as you entered the auditorium. If you have a question for the panelists, please write your question on the card, and we'll be collecting them on the side of the aisle, and then they'll be given to the moderator. So now let's get this symposium started. The first panel is Accelerating Cures and Advancing Human Health with Gene Editing. And the speakers are Dr. Corey Levine, who is a cardiologist and physician scientist at Washington University School of Medicine with clinical expertise in heart failure and cardiac transplantation. His research interests are focused on identifying new approaches to recover the failing heart and to improve survival and quality of life following heart transplantation. and Dr. Dongsheng Duan from the University of Missouri School of Medicine, Department of Neurology. And Dr. Duan's major research interest is to develop an AAV-based gene replacement and gene repair therapy for Duchenne muscular dystrophy. In regards to gene editing, the Duan Lab has recently demonstrated AAV-mediated dystrophin editing and muscle stem cells in vivo. So now I invite Dr. Levine to come up. Thank you.
All right. Got it. Thank you very much. Thanks for the introduction and the opportunity to come and talk a little bit about what we do today and share with you what we do on an everyday basis with gene editing and how that really affects patients. So what I was asked to talk about today is really what we do in and out clinically and how that extends into the laboratory and ways we're looking to the future to say how can we address these problems, how can we help our patients. And so I'm going to talk about gene editing and this concept of precision medicine or precision therapy, which I'll bring up in the context of heart failure. So as many people probably know, heart failure is a really prevalent disease. The incidence is very high. New diagnoses are very high. 5.7 million people are diagnosed annually 5 years ago, and as the population ages, that continues to grow. 6.5 million people were diagnosed between 2011 and 2014. And just a few numbers to highlight that. 14.9 million people in this country are affected by heart failure. 1 in 5 people, by the time they're age 40, will experience heart failure sometime in their future. Heart failure is 2 to 3 times higher in likelihood to cause death than cancer. And it accounts for almost $100 or $110 billion a year of spending. So it's a major problem from both a health and survival standpoint, as well as health economics. This is not just anywhere in the world, but also in Missouri. So Missouri, the darker red it is, the more prevalent heart failure is. And in Missouri as almost transplant, it's right there, so I know where it is. And it's really prevalent here. Not quite as bad as Texas and Oklahoma, but pretty prevalent. So what is heart failure? So most people, when they hear about heart failure, they think about a dilated or an enlarged heart, and that's really all it is. So it's a heart that grows over time because it's not functioning appropriately. When we think about causes of heart failure, we divide it into 2 categories. Ischemic cardiomyopathy, which all that means is heart failure due to coronary disease or blockages in the blood vessels, things that people get stents for or bypass surgery. Or the other category is called non-ischemic cardiomyopathy. Non-ischemic cardiomyopathy makes up more than half of the total diagnoses. We think of very rare causes that you hear about, such as viruses or toxins, exposure to chemotherapy, recreational substances. But the majority of cases are related to this diagnosis called dilated cardiomyopathy. Until recently, dilated cardiomyopathy, no one knew what caused it. So we said it was idiopathic or unknown. And in the past 10 years, amazing revolution in genetics has taught us really that this is a genetic disease. And we know that it's actually a disease of many genes. That's what I'm going to talk about. So with the advent of next generation sequencing, which is commonly used both clinically and from a research standpoint, dilated cardiomyopathy is due to mutation in many genes. Over 50 genes have been identified as causative for this form of heart failure. The genes, as you know, make proteins, and the majority of proteins that are affected in dilated cardiomyopathy affect the structure called the sarcomere. So the sarcomere is the molecular motor that allows the heart to contract. That's within the cardiomyocyte or heart muscle cells. So if you think about the heart as either a string cheese model, or if you're a barbecuer, like brisket, if you made it correctly, it can be, it takes many times to figure out how to make it correctly, but I've learned now. But you can almost peel it off, right? And when you peel off a piece of muscle, that's a muscle fiber. And then within the fiber are smaller pieces called fibrils. And within the fibrils are individual cells. And what makes the muscle actually contract is this thing called a sarcomere, which lives in individual cells. And it's composed of 2 sort of filaments, a thick filament and a thin filament, that essentially just slide over each other. And there's genes or proteins in between that act as molecular motors that grab on one and pull it. And that contraction is all based in the sarcomere. And the genes that are highlighted in bold are genes that we know cause heart failure that are mutated and present within the sarcomere. So we have an idea why many patients have heart failure due to dilated cardiomyopathy, but all the gray genes are genes that have been implicated with the disease that are not part of the structure. So we have so much more to learn. What this has taught us is a disease that we thought was unknown. We then learned it's genetic, and now we learned that there's more than 50 flavors of it, right? And so each person is a different person. And that is the concept of precision medicine. So if you walk into an average cardiology clinic today, and you have heart failure, like if you imagine every person in this room, no matter what your cause of disease is, we treat you the same way. We give you the same medicines, and we would say on average, this would be your survival. We think this would be your response to therapy. The idea that we can diagnose very specific causes of disease gives us an opportunity to tell an individual, this is what we expect is going to be in your future. This is what we expect your response to therapy will be. And this is maybe in the future we'll have a therapy specific to you. And that's what we're moving to. And that's the concept of precision medicine, which is treating a patient in a patient-centric standpoint rather than treating a disease. Does that make sense? And that is where all of medicine, for the most part, is moving to. And so just to highlight this in a few sentences, the promises of precision medicine are based on the fact that many patients with heart failure carry a genetic mutation, and that mutation can be used to flag different groups of disease. And it raises the possibility that mutation-targeted therapies or medicines can be developed to prevent, cure, or treat disease. And that's what we set out to do. However, there's many challenges till we can get there. How do we know that a newly discovered mutation is responsible for heart failure, right? We know these are associations, but we have to show that they're causations. How do we predict the natural history of an individual's patient's disease knowing the genetics? How accurate are we? And how much does it change from person to person who carries the same mutation? What's the best approach to efficiently develop patient-specific therapies? And can you actually group patients together and predict who will respond to one therapy and who may respond to another therapy? And these are the areas that we all are actively exploring and are making a lot of progress. So we're able to develop systems, which I'll share with you, that allow us to say that this mutation truly causes disease. We're able to use our patient biobanks and clinical registries to predict the natural history of patients' disease by genetics. And current group work in my laboratory and many others is focused on identifying specific therapies for individual forms of heart failure. And this isn't just a pipe dream. This idea works. This is a recent paper in the past year. There's been another two in the same topic, looking at cystic fibrosis, a really fatal and deadly disease that affects the pancreas and lungs. And a group came up with a specific small molecule that's able to rescue a mutation that's most common, this mutation right here. And when this is given to patients who have established disease, they're able to live longer, breathe better, and stay out of the hospital more. And so this is a reality and something that we can clearly achieve. So I'm going to give a real-world example of this. So this is a patient that I take care of. This is her right here. She's 28 years old, has heart failure. She has a long family history of it. It's a family affair for her. Her grandfather died of heart failure. We transplanted her mother, and we transplanted her uncle many years ago. Her mom died of heart failure after her transplant about 12 years ago, and she's really lived on her own since then. She herself had 2 heart transplants, and her brother actually lives with her uncle that she doesn't really interact with very much, and she's really been on her own throughout this, so she's a really strong person. And she says in clinic one day that her brother has moved in with her and is acting funny, doesn't know what's going on with him, and suspects that he has heart failure as well. We bring him into clinic, and that is true, and she says, well, what are you going to do about it? What can you do about it? What are the options? Why does this affect my family? So we went ahead and we genotyped her and her brother and her uncle and found that they had a mutation in this gene called lamin A. Lamin A, I'll show you, is a structural gene that is within the nucleus of a cell, and it's known to cause heart failure, muscular dystrophy, a form of muscular dystrophy, progeria, which is early aging, and forms of lipodystrophy, which is lack and abnormalities of adipose tissue or fat. This is what the mutation, her mutation looks like in cells. So the mutation causes the protein still to be made, but the protein's abnormal. And that's really the most common situation in heart failure, that the protein's mutated, protein's abnormal, but still present. And so this is a wild type cell, and it's in green, it's in the nucleus of the cell, and it wraps around it like an envelope. And her mutant still has a nucleus, but you can imagine it looks kind of shrunk and shrivelly, and the protein's still present. So we wanted to know more about why her mutation causes heart failure for her and what we can do about it. And so the way we can do this is a concept called gene editing, and this is what we brought up. Just to give some analogies of what gene editing is, this is a nice picture for it. We don't actually use forceps to grab a piece of DNA and shove new DNA in there, but that's in essence what we're really doing. So the way it works is we use an enzyme that acts as a molecular scissor, or cuts the DNA. And we can bring that enzyme or scissors to the DNA using a thing called a guide RNA. And all it really is a guide. It guides the scissors to a sequence of the DNA that matches the guide, right? So we can bring it to a certain area. That cuts the DNA, and then our body has ability to repair its own DNA. That's why our cells don't die when they're exposed to UV light or other sorts of toxins. And so our body can repair it in multiple ways, but if we provide a donor piece of DNA, right, that is the template of the new sequence we want to put in, well, the body will, or the cell will naturally repair such that we actually did this, where we made a very targeted and specific substitution. And we can use this to create patients' mutations, or you hear later how to correct patients' mutations using gene editing. And Cas9 or CRISPR is the name of the system we use. So maybe I should use the title Mo CRISPR. I like that a lot, actually. So, but that is an essence of what we're doing. And so our lab does this in two different ways. We use zebrafish, which is a vertebrate small organism, to study heart failure in a high throughput standpoint. And then we use human cardiomyocytes that are engineered into tissues. And I'll show you an example of that. So we make patient mutations in zebrafish. Zebrafish are a great organism to work with. They're optically translucent, which means you can just watch. You can observe what the heart does in terms of development and function. Their sarcomeres, that building block or molecular motor of the cell, is almost identical to the human. In fact, at the protein level, there's 90% identity in many of these genes or proteins. And within the areas where there's mutations, it's almost 100%. So it's really the human protein in its native environment. Zebrafish are great because you get hundreds of eggs every time two fish mate, so we have a robust sample size. And they're very amenable to using this sort of genetic manipulation. So here's an example of what it looks like. This is a zebrafish embryo 96 hours after fertilization. The heart's this little guy here. And this big sac is called the pericardium, and it's very swollen. And it's swollen with fluid because the fish has heart failure. And we've used this organism to knock in a whole bunch of mutations that are commonly found in patients. This is Whitney's, or that patient's mutation right here. And we use the fish to try to understand why it has heart failure, to understand if there's structural problems, what's the deal with conduction, is conduction abnormal, and to try to figure out whether there's interactions between different mutations. And this is an example of what this looks like. Just put a picture with some words. So this is a zebrafish heart. This is the atrium or top chamber, so blood comes in here. It goes through this valve into the ventricle, and then it goes out this area right here, which is equivalent to the aorta, the large artery of the body. And then this is our patient's mutation. And you can see the heart's small. It contracts abnormally, doesn't squeeze well, and it's actually beating in an irregular way. And it actually looks just like her heart before her first heart transplant. Okay, and what we're doing with these systems is we screen libraries. We start with libraries of drugs that we know that interact with our gene or protein of interest. We get candidate drugs and targets. We, instead of sometimes looking at contraction, we've made an assay where fish that have heart failure emit a lot of light as a reporter, and we look for compounds that improve the function of the fish heart. And those compounds would be potential therapeutics that'd be specific for a specific mutation. So that's great for fish. How do we do it? How do we show that this compound will work for people, right? You just don't want to go into a person. And so the way we do this is using another gene editing technique. And this is used something called induced pluripotent stem cell derived cells, and we can make little heart tissues. So we take samples from patients, from skin, blood, or urine, and then we have some cells. We can then reprogram themselves and edit the genome to make them stem cell-like, but not stem cells. And we can engineer different mutations into them from our patients. And then we can coax them into being cardiomyocytes. But to study them, we want to make them heart tissues. So then we can assemble these cardiomyocytes on an apparatus where they form what we think look like normal cardiomyocytes for humans. And we can watch this apparatus beat. And so it's on 2 poles here. So this is during relaxation. The poles are straight. And during contraction, they kind of bend in. And we actually can watch it and measure. And this is an example of what it looks like. So this is a control cardiomyocyte tissue. And you can see these poles kind of moving in every time it beats. And this is an example of a different patient's mutation engineered into cardiomyocytes in a tissue. If you think the video stopped, it's because it's not beating well. It is beating, but it's beating extremely slowly, dysynchronously, and poorly. And so but the cardiomyocytes are still there. They're arranged in a normal way. They just fail to contract normally. And so this is a great system where we can add drugs, and we've been able to add drugs that improve this mutation's ability to bind calcium that then make it look like this. And that's how, and then the next step is to go into people. And so that's one way where gene editing has been a great technology that's accelerated our ability to learn about these mutations and why they cause disease within the heart, and to come up with therapies that can be translated to patients in a fairly short time frame, usually less than 5 years for small molecules, which is pretty good. So the take-home messages I wanted to share are that many diseases, including heart failure, have a genetic basis. You can identify relevant mutations, and that teaches us why disease occurs. It is essential for this transition to go from one-size-fits-all medicine to precision therapy where we're treating individual patients. And there's incredible promise of precision medicine, but it's really limited by our ability to decide which genetic mutations are relevant and to develop strategies to efficiently generate therapies that work for an individual mutation. And genome editing represents really an effective solution to overcome these hurdles and translate our knowledge into applications. And this era is really here. It's here throughout the US, it's here in Missouri. If you walk down the halls of Washington University where I work, almost every lab there uses this sort of technology in their own diseases, in their own ways. And the amount of innovation and transformative work has just exponentially increased over the past 5 years. And I think these are great technologies and solutions that improve the health of our patients. So with that, I want to give you just a glimpse of the number of people it takes to do work like this. This is the real people doing it. It's a group of senior scientists, postdoctoral fellows, people after their PhDs, people that are graduate students, even people that are undergrads and high school students have been involved with this research and are able to contribute, as well as all the important federal and foundational funding that makes this possible. Thank you.
Okay, my name is Dongchen Doan, and what I want to share with you today is some of our experience in trying to use this fancy CRISPR technology.
Can you use the microphone?
Oh, micro. Somewhere. Still this. Hello?
No.
No. I need to have a quick therapy.
Hello?
to my
Hello?
Voice to get it louder. Great. It's fantastic therapy. Worked. So we're going to share some of our experience trying to use this new technology to deal with a disease called Duchenne muscular dystrophy. And here's my disclosure with the company that is interested in developing therapy for muscular dystrophy. And so first, what is Duchenne muscular dystrophy? I mean, for many people, maybe it's something I have never heard about that, but I can share a story of my friend. His name is Harvey, and this is when he's 4 years old. And this is how he grows up. Now he's 12. This is a disease called Duchenne muscular dystrophy. If not treated, this is on the outside, is a patient from India that has never received any treatment at 18 years old. And so this is a disease affect all the muscles in the body. Skeletal muscle, cardiac muscle, you will end up become scoliosis, lost your working ability, you die from cardiomyopathy or respiratory failure. So the disease was named after a French physician, Duchenne. He described the paper back in 1868. And the gene that is responsible for the disease was cloned by Leconquer's group in Harvard back in 1987. And they call this gene dystrophin. So what does dystrophin do? And here I'm showing is a normal cell, and this is muscle cell membrane. Your muscle cell is embedded in the extracellular matrix. And so when you exercise, your muscle cell is going to change the shape, right? That's different from liver cell, your brain cells. It contract and relax. And so what we need is to have those something to hold it together, and this is dystrophin. If you look using a fluorescence microscope, using a marker for dystrophin, you will see there are kind of stand around the cells, and the muscle looks quite healthy. And this is what happens when you lose your dystrophin. Your muscle eventually undergo degeneration and necrosis, and you don't see any dystrophin, and your muscle is hugely a big mass. Inflammation, fibrosis, and big cells. And this is what we're trying to do, that what our hope is trying to remove the mutation with the CRISPR, and then to get a dystrophin which is slightly different from the wild-type dystrophin, but hopefully we're going to get the disease symptom reduced for the patients. So how can CRISPR be used to treat, to restore dystrophin? So this is a kind of what I showed that. Let's say here is a mutation in patients, and so what we do is, so we use a guard RNA, okay, which is kind of marker. It marks where the mutation is located, okay, and then we have the second component of this CRISPR machinery. It's an enzyme like scissors. It will going to cut where exactly this guard RNA points at. And then, after this bad part is removed, you get a shorter gene, but still functional genes. So you get a dystrophin back. And so then I can show you some of the work we did with mice. So this mice is called MDX mice. It has a mutation in the exon 23, which is a stop codon there. And basically, your dystrophin will not be produced. You don't have protein. So what we do is that we use this AAV, or almost a virus, almost not a virus, a nanoparticle, a bio-nanoparticle. So we put our Cas9 into one of the AAV vectors and guard RNA into a different AAV vectors. Then we put them together, deliver to the mice. And we have a guide with direct where to cut, and scissors will cut around this mutated exon 23. And after remove the mutation, you get a inflamed protein will be produced. So this is an example that we showed that by directly injecting those AAV CRISPR virus or vectors into the diseased muscle. And this is normal muscle. This is untreated muscle. You see the muscle degeneration, necrosis. You see a lot of macrophage and neutrophil. Those stands for inflammation. And this is after treatment. The morphology much improved, and inflammation was much reduced. And also, we looked at muscle force. And without treatment, it's lower. After treatment, it went up. And this is an assay which we test, stress assay. And the lower, the worse. the higher the better. So after CRISPR therapy, we also see improvement. And, but that's a direct injecting to treat a single muscle. I mentioned that disease is affect all the muscles in the body. So you have to have a method to get all your vectors to all the muscles. So this is what we did. So we inject those CRISPR vectors into tail vein. Then it goes through a body. And this 3 months later, we look at those. We can see that in multiple different muscles, even including heart, we can see the restoration of the dystrophin. And it's suggesting it is work. And at the same time, we published in collaboration with Charlie's group and also 2 more groups, Eric's group and Amy's group, showed that, yes, you can use a CRISPR to treat a disease, a human disease in the mouse model. But I want to emphasize here, short term. And this is another example trying to use CRISPR to treat mutations in the human patients and in the dog model. So in this case, it has a mutation that exon 50 was removed. This removing of this exon 50, so exon 40 would not be matched with 51, so it creates an out-of-frame mutation, so you will not be able to produce protein. So there are 2 ways to get back with CRISPR. One is the one I just showed. You can have 2 CRISPR-Cas9 cut and flanking this additional exons, which is there. But if I remove this, then 49 can get to 52, and which is shorter, but it's functional. And alternatively, I can just use 1 Cas9 and modify its splicing profile and splice it out going this way. Or because this deletion, it introduced out-of-frame mutation by inserting a couple of actual nucleotide. So what I do is I just use CRISPR to cut here, remove this field, and then I will be able to get a exon 51 slightly different, but they're in frame. So this has been done by Eric's group, and they have recently shown that, well, they can get it done in dogs for at least 2 months. And this is kind of one of the biggest news in the last few months. But there's an issue with this. So I mentioned this is a lifelong disease. If you treat 2 months, 3 months, it's not going to work, right? You need to have long-term therapy. And we have a lot of concerns, about CRISPR long-term therapy. So we decided to take a look, address this issue. So what we did is that we used MDX mice. We delivered the CRISPR at 6 weeks, and then we waited for 18 months, then see what happens. So this is the first experiment we did, and we can. So we did exactly what we did when we did a 3 months experiment, short-term study. And what we see is that, well, after CRISPR therapy, we can get some, if lights can be dimmed a little bit, that will be more easy to see. If not, open your eyes. Oh, believe me. So you can see some edited cells, dystrophin restored in the heart. But in skeletal muscle, always this background, essentially there's nothing there. So it works for heart and not for skeletal muscle. And this is kind of a different way of looking at that. And this is where the edited dystrophin in the heart, in the skeletal muscle, if you look at quantification, there's almost nothing in skeletal muscle. So since there's some dystrophin restored in the heart, so we see if it can improve heart function or morphology. And this blue stands for fibrosis. You can see that the untreated and the CRISPR treated, they're the same. So essentially, the therapy doesn't work, right? So this is really frustrating because after 18 months, now we get nowhere. And then we went through, did more experiment. What we find is that a very intriguing finding is that CRISPR has 2 components. One is the guide that's pointing out where to cut. One is the scissors that's cut, right? When we look at those 2, we find out that all the vector genomes that Cas9 we have plenty, but the gut RNA was reduced dramatically, and in skeletal muscle, it's essentially gone. So this is suggesting that for the reasons that we don't understand, when you try to deliver the gut RNA by itself, it's going to get selectively degraded for a mechanism yet to be understood. So okay, say if we say, well, if this is a problem, let's just put more gut RNA into the system, see what happens. And so this is what we did. Now we can see more in the heart, and we do see a very nice, dystrophin restoration in the skeletal muscle. And this is, again, Western blot. You can see in the heart, there's more. In skeletal muscle, you began to see them. Instead of in the previous study, you didn't get anything. So we did this study. First, we did in male mice. And then we look at the skeletal muscle fibrosis. So all those blue things are fibrotic tissues. And as you can see that the fibrosis was substantially reduced after CRISPR treatment for 18 months. This is a quantification, show the same thing. And this is looking at skeletal malfunction. And again, this is untreated. This is treated on twitch force, untreated, treated on titanic force, and this is untreated, treated on essential contraction. And clearly, there's still way to go to get to the, to the Y type, but we're seeing improvement. And so we also decided to do female MDX mice. From our past study, we find that female MDX has shown more severe cardiomyopathy. They show dilated cardiomyopathy exactly as human patient. Male mice don't show that. And so we did the same thing, introduced more RNA in the injection, and we see more CRISPR restoration of the dystrophin in the heart and also in the skeletal muscle. So again, on Western blot, you see the nice restoration in the heart and also in the skeletal muscle, and they're there's a range, but in generally, they're much better. And if you look at the cardiac fibrosis, this is untreated. You start to see the myocardial blue things there, and here it's almost gone. And this is quantification showing the improvement. So how about function? So we did catheter assay. We put a catheter into the heart of the mouse and measure the system volume and system volume and ejection fraction. Ejection fraction is a common, most people are probably familiar with that. You can see that this is untreated, this is treated. There's clearly improvement there. And I mentioned that Duchenne cardiomyopathy is a dilated cardiomyopathy. So their heart becomes larger. So at the end of systole, their volume would be larger. And this is after treatment. Their volume goes down. So we got very nice improvement. So we're very happy with this, and this paper is in press right now. So hopefully we'll help other groups to deal with this issue. And I guess I'm still not used to trying to keep going back to my mouse pad. And so the other question I got asked so often is that, well, you have a degenerative disease, right? And but your muscle have regenerative capacity. What if you can treat muscle stem cell with your CRISPR editing? Well, then all the regenerated cell will have the dystrophin in there. So this has been debated very hotly. Two groups. One group says yes. One group says nonsense, no. They try to fight each other. But the method they used, we don't like, because one group trying to do an in vitro manipulation, we find that may introduce some artificial problems. Or, the other group, what they're trying to do is they're trying to look in the microscopy to find those edited genes. And if you look at the entire muscle section, even in the normal case, probably find 1 or 2 stem cells. You're lucky. And if you're trying to find like 1% or 2% of those cells get edited, it's impossible. You can easily miss it. So we decided to totally take a new approach that trying to avoid all whatever people have been doing. So what we decide to do is that we do a CRISPR therapy, and then we take the muscle out, and we graft it to a different mice. And this is what I mean. So at day 0, we have green mice. We take the muscle out, this green, and we graft it to a white mice. And in less than 1 week, all the green muscle will undergo necrosis. And this is what you can see that. This is host muscle, and this is 1 week, and the graft, they all die. The only cell that can survive those nasty, environment and this ischemia is the muscle stem cell. And then we say, okay, let's wait, see that come back. And when they come back, if the stem cell from the host contributed to the regeneration of the graft, you'll get a white muscle back. If the green muscle contributed, you'll get a green muscle come back. Okay, so here's the experiment, what we did. And first, we grafted the green muscle mice to white, and white was labeled with yellow here, and green. And we can find almost nearly 100% of those regenerated graft are green cells, suggesting that it's indeed from the donor. And we also did an opposite experiment. This time, we put the green into the host. We want to see how many cells, if we look at the regenerated graft, how many of this green cell will be in there. We find less than 3%. So very few causes contributed to this regeneration. So this is trying to say that, well, whatever comes with this graft, when they regenerate, it's have to come from the stem cells that have been edited or treated. So this is the experiment we did. So we treated mice, waited for 18 months, and then we take muscle out. Some of those we quantify dystrophin, see what's going on before the grafting, and then some part of that we grafted. And before grafting, apparently, with the dystrophin, with CRISPR therapy, we're seeing that improved the dystrophin expression about 20 to 30%, which is within the therapeutic range. And so this is after graft regenerated. Okay, so I want you focus on here. So these groups, they were not treated with CRISPR before they were grafted. And this group, they were treated with CRISPR before they were grafted. Clearly, the group treated with CRISPR before has more dystrophin positive cells. Okay. And if there is CRISPR editing there, you should see edited genome, right? So we decided to look at that, and so this is what we did. Oh, sorry. So this is what we did. So we designed those 2 guide RNA to cut on this side. Then if we do PCR, we'll be able to detect these fragments, which should be the edited genome. This is before grafting. So we see that with CRISPR treated, we see this band. Without treatment, we don't see this band. And this is regenerated graft. We don't see that. And with the CRISPR treated, we see this band. And then we did another round of nested PCR to kind of amplify signal. And here, without CRISPR treatment, you have no edited band. And after CRISPR treatment, you have the band. And this is before you grafted muscle. And this is after you graft and muscle regenerates. And without treatment with CRISPR, you don't get anything. With treatment with CRISPR, you do get an edited band, suggesting that that's indeed the genome has been edited. And this is sequencing showing that, now this is before grafting. This is what we see the ligation of 2 parts that was left over from the cutting. And most of those is perfect. And this is the clones from the regenerated graft. Again, we get very nice molecular evidence. So, so in summary, we have shown that a single AAV9 CRISPR therapy can lead to therapy for long-lasting, up to 18 months. And those mice, if treated, their lifespan is 20 to 21 months. So we're almost pushing to the end of their lifespan. And also we showed that in CRISPR therapy we're doing, we can get to the muscle stem cell. And I want to say, it seems I'm trying to portray a beautiful future for the CRISPR, but it's not the whole story. We all know there are other issues, potential immune problems, and even with some of the published data, people are arguing, right? And whether your CRISPR, your Cas9, your enzyme there, hanging on there, is going to cut, eventually can cut something, right? So there are a lot of issues. So we believe that this technology is still at its early stage, but it has a future. And so the work was in collaboration with Charlie Gersbach's lab at Duke. And the CRISPR 18-month study was mainly done by Shadi Hakim, Dr. Shadi Hakim, and Dr. Nalina Wosla. And Michael Nance is an MD PhD student. He did that stem cell study. And our research was funded by NIH and the Department of Defense and Hope of Javier. And this is actually my friend and his mother says, well, we need to fund your research. We want you to do it. And the boy actually, we are Missouri, right? We're Columbia. We are tigers, right? The boy manually made a tiger for me. I put that on the desk of my office. It's just really, really goes to, I see it every day. I know this, we have to do something. Thank you.
Can you hear me? Can you hear me? Dr. Dewan and Dr. Levine, thank you so much for those presentations. And gene editing is a fairly new type of cutting-edge research. And so I'm going to ask you all to kind of look in your crystal balls, look in the future, like 20 years down the road. I mean, what are the potentials for gene editing?
I'm always positive, no matter what. Even at the worst time when people say gene therapy is not going to survive, I say, no, that's my career. I have to go. Still, same answer. I believe in my career. I believe in my research. And I'm sure, there are issues that we're facing right now, but all those can be dealt with.
Yeah, just in my career, you probably can speak in similar ways. Gene editing used to take the most experienced person in your laboratory, even a core facility, to do it. And now high school students easily do it in the laboratory over summer. The technology has advanced just in the past 3 or 4 years to the point where it's really incredible. So if I look forward, how many years? 20 years. So right now it's already a lab tool that's used in all sorts of animal systems, ones where the genome wasn't previously accessible. You'll hear that's used in plants, I'm sure, shortly. So that's where it is right now. I think in very short periods of time, you're going to see it used in diagnostics laboratory tests for patients within clinical laboratories, maybe within 5 years or less. And then I imagine it will be used in a set of diseases therapeutically that can be corrected, especially in tissues or organs that are regenerative like muscle, to correct specific mutations. I think we all recently heard some of these interesting studies curing blindness, which is one of the first CRISPR-based therapies to cure disease and restore vision. So I think the future is very bright.
And I know that there are concerns, or people want to make sure we're doing it in a thoughtful way, and the NIH and FDA being involved. And I had the opportunity in May in DC to hear FDA Commissioner Scott Gottlieb speak about this, and he's very excited about it. But what exactly are, like, NIH, the funding for gene editing, or certain guidelines? But I know that the United States is moving forward in a very thoughtful way with this type of research.
Well, CRISPR is not only limited to therapy. I mean, as you will hear later on, it's affecting every part of your daily life. So this is an important technology. And let's say my lab also working to develop a gene replacement for the Duchenne muscular dystrophy. And that we started in 2002. And last year, we have first human trial. So hold on. In general, people said it's going to take at least 20 years to go from the scratch, from the salts, from your bench, eventually move to the bedside. So there are excitement there. That's why there are huge potential there. That's why NIH want to fund it. That's why patients want to support it, sponsor this research. But there are also issues, as I mentioned, that Cas9 itself is derived from bacteria. You don't want a bacterial protein to continuously stay in your body, and that's going to cause immune problem. And eventually, your body immune system attack is going to kick out Cas9, kick out the treated cells at the same time. And the other thing people were saying, well, you have this Cas9, they're just an enzyme. At the beginning, it goes to wherever you guide it to. But over time, this is not like a high fidelity or super high fidelity thing. They get tired. They need sleep time. So they do their sleep work. And that's where the bad thing happens. So there are many, many more things need to be dealt with. That's why the NIH and FDA and the many major industry, they want to invest in this technology early on. So hopefully, if we all work together on different aspects, and we can speed up this process, hopefully 20 years will be more, we see something, some product. And the CRISPR therapy itself, per se, there's a couple of clinical trials has been initiated in United States. But in all those trials, people that will take patient cell, take it out, then treat it in the petri dish, then they screen it to make sure that the Cas9 is gone, and they don't have any mutations introduced. Then from the screening, they find one cells that's good, a few cells that's good, and they put it back. And this is kind of more like stem cell. This might work for some disease, but definitely not for skeletal muscle. I mean, how many cells you will need to fix all the muscles in the body? Think about it.
I think to answer some of the regulatory questions, I think from a research standpoint, it's very well supported by governmental foundation support. And obviously, it's also within startup communities. From the FDA level, the regulation is actually quite high. So what the FDA regulates is the target of the will regulate the target and the technology. So the targets of genome editing are the disease and the mutations themselves. They're not things like hair color or physical features. So the FDA is highly regulatory and really is very careful about making sure that this technology is developed in an appropriate way where it's only fixing the disease and there's nothing more to it. So I think the FDA will keep a very close eye on this. The scientific community itself is very good about policing its own sort of research and its own goals.
Well, again, I want to thank you both. Your presentations were absolutely fascinating, and this is such exciting, cutting-edge research. So, again, thank you.
Thank you.
Thank you.
Okay, we're going to transition here for just a second. I know that...
So we're going to be around, both of me and Dr. Levine, and if you have questions, feel free to challenge us.
And I think we're going to put another chair on the stage for the next panel. Is that correct? So, okay. Thank you. Okay, so the next panel Panel 2 is developing a more healthy, sustainable food system through gene editing. And I'm going to introduce the moderator, Dr. Shauna Lemke. She's the Director of Food Strategy and Stakeholder Engagement with Bayer U.S. Crop Science. Shauna?
It's not on yet. Oh, there we go. Okay, thank you. Good. Well, good morning. Thank you for the introduction. I'm so pleased to be here and very pleased to be hosting our next section of the program, which will be more of a panel discussion session. We won't have any slides to share with you all, and that's going to be on food and agriculture. So it was really, really great to see the work going on in medical science. I think it really speaks to the power of when you have innovation and tools and technologies like this, how you can have really great benefits across a number of different fields. So before I introduce our panel, I just want to kick us off with a couple of introductory comments to sort of frame the discussion for you. So speaking personally, I've been involved in agriculture virtually my entire life. I grew up in and around farming and then ended up as a career in agriculture, working 13 years with Monsanto in town, and very recently joined the Bayer Crop Science team. My educational and science background is in food safety and nutrition, so I'm really pleased that part of what we'll discuss today will get at some of those kind of human health, nutritional benefits, and sustainability as well that we think this technology can offer. So as we start thinking about agriculture in this section, there are a couple of themes I just want to prime you for a little bit. First of all, I think you're going to see a lot of excitement about the potential for farming and for practitioners of farming with this technology. I think growers tell us all the time, every year, that they're looking for more and more tools that help them with their day-to-day challenges. So that, they're dealing with wanting better quality product, dealing with weather, and they're dealing with challenges like disease and weeds and things like that. And so we think there's a lot of benefit that can come there. But then there's a second theme here of benefits beyond the farm. And so we're going to talk a little bit about sustainability, the fact that these tools have the potential for us to look at bigger picture problems, like limited natural resources. I think there's also a keen interest in connecting with the consumer on these products. I was listening to a news story this morning, again, talking about the number of people leaving the farm. And so we're at a time now where very few people touch agriculture, but conversely are very interested in knowing about their food, where it comes from, that it's being made in a healthy and sustainable way. So we definitely want to talk about that. Again, I think I'm personally excited about some of the things we've seen discussed in terms of applications for gene editing. Many researchers and companies have talked about bringing products that have a nutritional benefit. So thinking about better soybean oils or low gluten wheat as a couple of examples. So finally, I think the third area we really want to highlight for you all today is, again, as Dr. Carrington kicked us off, the importance of innovation in this area for Missouri. So you'll be hearing from people who are based here. I represent Bayer, where we're doing work both in healthcare and in agriculture, and the agricultural work happens here in St. Louis. And so I think all of the panelists, want to share that story with you as well today. So what I'd like to do is invite my panelists up, and then we'll kick this off with about 40 minutes of Q&A, and want to make sure everyone in the audience also is thinking about your questions. We'll try to make time for that in the end as well. So first we have Dr. Natalie DiNicola, she's the Chief Communications Officer with Benson Hill Biosystems. Next, Dr. Randall Prather. He is the Curator's Professor for Reproductive Physiology and Molecular Biology at University of Missouri-Columbia. And finally, Ms. Catherine Wojcicki, who is the Assistant Director of Communications for National Corn Growers Association and a family farmer. Are these on? Okay. Can you hear me? Okay. So welcome, everyone. To kick us off, I'd like each of you to kind of share a little bit more about who you are, what your work is, and how you connect to this area of gene editing. So now let's start with you. How is Benson Hill investing in gene editing technologies, and what sort of innovations do you think are needed to contribute to a healthier food system?
Thanks, Shauna. And good morning, everybody. It's great to be here with you today. A little background on me. I'm an environmental toxicologist. I have worked in agriculture and food for about over 20 years now, largely because there are very few industries that can have as much impact on conservation of our natural resources and environment, health of our planet, and ultimately ourselves as food and agriculture. And I serve as Chief Communication Officer at Benson Hill Biosystems. We're an early stage company that actually was born out of research that was done right here at the Danforth Center, really trying to harness a lot of the advances in computational science, big data analytics, machine learning capabilities, and marrying that with real understanding around plant biology and particularly plant genetics, so that we could build a platform that we call Crop OS, Crop Operating System, which is really intended to enable all different kinds of companies and organizations working across the food and agriculture value chain to improve crops, to build a more healthy and sustainable food system. Some of that work can be through traditional breeding, where essentially we'll take a couple of two different varieties of a plant, where there's certain attributes we like in each one, and we combine them together to try to get the right combination of the particular attributes we're looking for. This data science capability allows us to do that faster than we used to be able to. We can make better predictions of which two plants to genetics to combine, but we're still combining the entire genome of the plant. So it takes a lot of time, a lot of rounds, a lot of planting, and frankly, quite a bit of money to do that kind of research. Gene editing is another tool that we make available, and just like you saw in the medical community, it's really allowing us to better understand what is the particular gene within one variety of the plant that we're really going after for a particular attribute, so that we can basically copy the way that's being expressed in one variety in the other variety that has the other attributes we're looking for. So by doing that, it allows us to make those improvements much more precisely and intentionally, and get to the outcome that we want. So why is that so important for food and agriculture? Because the traditional breeding, because it took so much time and frankly so much money to do that, you only had the largest companies focused on that, mostly seed companies who were very focused on the largest crops out there, namely corn, soybeans, and cotton, which are very important crops, and mostly focused on the benefits that directly for the farmer, particularly around productivity of those crops, which is absolutely essential from a sustainability perspective and, of course, the profitability of those farmers. However, there are 30,000 edible plant species on our planet. Our food system relies on about 30, and almost all of the genomic innovation, the genomic improvement, has just happened in those three largest commodity crops. In fact, 2/3 of our calories around the world are based on 5 different commodities. So there's this enormous amount of diversity of different crops out there and diversity within each crop species that we really have not been able to harness to date as effectively as we want to. And consumers are really wanting to see a lot of improvements in our food system in terms of more diversity, more focus on sustainability and better use of natural resources, more focus on health and nutrition. And through gene editing, we can make these kinds of improvements that directly benefit farmers, but also that directly benefit consumers at the same time in a lot of different crops, so that you can see less bitter specialty grains, less bitter leafy greens, more drought-tolerant crops out there, just removing allergens from food. Lots of different improvements that are benefiting both the consumer and the farmer at the same time. And that's why Benson Hill, as a platform company, we're really excited to try to empower that kind of innovation through a lot of organizations that are really committed to try to continue to improve our food system.
Thank you. So advances in gene editing aren't limited to plants. Dr. Prather, could you talk a little bit about what challenges in animal agriculture you think there are opportunities with this technology?
Sure. So I'm Randy Prather. I'm at the University of Missouri Division of Animal Science. My area of study really is early embryo development, from one cell to blastocyst for about a week, and I study it in the pig. And because we study that area of development, that allows us to then go in and make genetic modifications to one cell stage embryos and then study the effect those modifications have on development. There's about a 30% loss of conceptuses during the first month of development in most all mammals, and hopefully we'll be able to identify where some of those losses are occurring and have that application also to humans as well as agriculture. But because we can go in and make these genetic modifications, the question is, which ones would you want to make? Well, as you look across agriculture, pigs more specifically, there's a disease out there that doesn't have a solution, and that's called porcine reproductive and respiratory syndrome virus. And it infects pigs, and the costs in North America and Europe are about $6 million a day. The vaccines don't work. That was $6 million yesterday. That's $6 million today. That's another $6 million tomorrow, and that does not include Asia. There's no solution out there except for gene editing. We've gone in and used a CRISPR-Cas9 technology to actually remove a protein called CD163 that the virus uses to infect the cell. So now the pigs don't get sick, they don't get infected. Okay, so some problems that we have in agriculture only have a genetic editing solution. There's other things that we can address. We can address things like introducing a gene from Celtic cattle or Angus cattle into Holstein cattle that removes horns. Well, that's an animal welfare issue. We can also look at changing the composition of the meat so it's more healthy. There's a whole range of things that can be done with gene editing and genetic engineering in production agriculture, in specifically pigs, that in some cases there's not a solution.
Thank you. Catherine, give us a little bit of the grower's perspective. What are your hopes for what this technology can do?
Sure. I am with the National Corn Growers Association, and I've worked in agriculture for 11 years. But I grew up in Clayton and was not familiar with farming. 5 years ago, I married a farmer, and now I'm extremely familiar living on a farm and helping and working, as everybody really has to. From my perspective, I come from the Metro East. I'm just going to admit that at this point. We live in Illinois, but I work with a lot of farmers in Missouri, and I talk to them too to make sure that I'm correct about this. Some of the biggest challenges we're facing in Missouri, and on the other side of the river as well, are challenges related to climate change and things like drought, which was faced in Missouri this year again to a pretty extreme level. Things like invasive pigweed species, Palmer amaranth, and water hemp, and also things like soil erosion that we're hoping can be addressed through genetic editing, providing us with new tools in the toolbox that will allow us to reduce tillage and to improve our soils so they can be healthier. I really think that farmers and consumers have very similar wants in a lot of ways because we both value having healthy soil and clean air and clean water. And so I think a lot of these tools offer a promise of not just providing more abundant or more affordable food, but also helping us care for the planet we all love.
Well, that leads me actually to my next question, which was around sustainability. So this is an area I think consumers, they've heard the word, not always sure what it means. I think generally there's a desire to know animals are well cared for and that we're making the best use of land and water and soil. Question to anyone in the group, what else do consumers need to know about the possibility for sustainability benefits with this technology?
So as I look at PRRS again, I'm going to keep coming back to that because that's really a great example. I think a lot of times we give lip service to sustainability. If we're really going to be serious about being sustainable, then if we don't introduce some of these edits and correct some of these problems, at least for PRRS and pigs, we're assuring that we're going to continue to provide food, fuel, labor into animals that are going to underperform and get sick. If we're serious about sustainability, we need to incorporate these technologies so that we address those issues. I think another one that consumers are concerned about is animal welfare. Nobody really wants to watch animals get sick and die. Producers don't, consumers don't. We have a solution for this disease. We could get rid of it.
Anyone else?
I would say one of the biggest things I think with sustainability is when you look at the labor-intensive practices that are necessary for some forms of agriculture, people don't necessarily see that things like GMOs and gene-edited crops can actually make farmers much more sustainable by reducing the amount of times that they have to drive large equipment across something, and therefore, too, the greenhouse gas emissions used in the production of crops. They can build up the soil and reduce things like runoff and keep things right in place where they need to be instead of having to over-apply and hope it stays there. I think that also there's really an exciting possibility with all of this to be able to improve soil health, which is something very important, I know, to Bayer and to NCGA and to farmers everywhere. And I think that by having the ability to take care of the land even better, farmers will be happier, and I think it's good for consumers as well.
And maybe just to build on the climate angle as well, it's not just about using all those resources more efficiently, like water, less greenhouse gases, things like that. But it's also helping our crops be able to adapt to a changing climate and the heat intensity. One of the really exciting projects going on right now is around chocolate, which is grown in tropical environments where there's pretty significant change in climate happening. And one of the things that's being done with gene editing is to try to make the crop, the tree, the cacao tree, more resilient, more heat tolerant, because what the farmers are having to do in these different parts of Africa is actually go up the mountain to plant the tree higher and higher, and in doing so, they're deforesting the mountain as well. So really trying to help our crops adapt better is really critical. There's another, though, angle of sustainability, not just about the farming itself, but actually in the processing of some of our foods. So quinoa, a really exciting crop, nutrient dense, highly nutrient dense, but it has a very bitter flavor component within it. And right now, what we basically do to remove that flavor component is we wash it out with lots of water. And that processing actually affects the sustainability profile of the crop. It also washes out some of the nutrients of the crop. And there are varieties of quinoa that don't express the protein for that real bitter taste, that gene for that bitter taste as much. And if we're able to actually knock out or deregulate, downregulate the expression of that protein in the quinoa crop that we're trying to commercialize, that allows us to skip this whole step in processing and improve both the nutritional profile and the sustainability of the crop, as an example.
Good, thank you. So I was talking about in my opening remarks about the consumer. I think, my perspective is we're at a time of great interest, but unfortunately, without sort of firsthand experience, terms like antibiotics or GMOs have become kind of hot topics for consumers and a lot of debate. So I guess as we think forward about bringing new tools into agriculture, how are you all thinking about that? How are you thinking about addressing and working with the consumer ahead of time to understand what we're doing?
I know at NCGA, working together with a lot of other trade associations that represent different parts of the value chain, like the American Seed Trade Association and some of bio groups of that nature, we've really wanted to get out and be transparent as possible. And so using some initiatives that bring together real farmers with real consumers, things like Common Ground, which is a program we have that brings together moms from the farm with moms from the cities to talk about what they have in common, feeding their families and what they care about. We've started talking about gene editing actually about 2 years ago on that platform, and really trying to answer questions, knowing that we still have a lot of questions, and we don't necessarily know what's going to come out first or when it's going to come out. But trying to say, we know this is coming, and so let's start talking about it. and let's address some of the questions people might have with animal welfare, and trying to open a conversation so that they know that farmers do care about what they think as well, and that we share a lot in common and want to communicate. So I think we're going to continue with a very proactive stance in trying to start a real dialogue that's more based in what we have in common and trying to find solutions that are good for everyone instead of just pushing out a message.
So I think with the example of CD163 and knocking it out, and we actually get rid of a protein, the pig genome has like 3 billion letters. And we can change 2 letters of that genome and get rid of this protein so that the pigs won't get sick from PRRS. During normal fertilization, there's probably 1 to 200 random mutations that occur. But for some reason, the FDA thinks we need to regulate when we do those 2-letter changes on purpose. I think if people understood really what we're doing is just changing a couple letters of the genome, we're actually getting rid of a protein, I think they would be more accepting of it. I don't know how you do that educational component. There's other gene editing that obviously is going to involve different things that may take some more regulation, but some of these very simple edits, changing a couple letters of a 3 billion letter genome, and we know what those changes are. I can't comprehend what's unsafe about that.
Well, just maybe to build on that a bit too is I think there's a lot of recognition that it's really about the oversight of the product, not necessarily the process. So really trying to understand the risk benefits of the kinds of changes that are happening. And what I see happening with the plant industry, at least that I could speak to, is a lot of outreach to all different kind of stakeholders across the entire chain, from farmers all the way through to consumers, conservation organizations, social justice organizations, consumer watchdog groups, if you will, all really trying to come together and have a dialogue, not necessarily education, because education sounds a little one way, but more of a two-way kind of dialogue of understanding what are the values people are looking for in our food system and how can a tool like this fit. What is the appropriate kind of oversight for the kinds of changes that are being made? And then what are the right kinds of best practices as an industry that we should be putting in place? And one of the things that's pretty interesting I see is in 2015, the amount of investment in food and agriculture actually quadrupled from about $900 million a year to about $4.5 billion. And a lot of that was coming from people that were not from agriculture traditionally. Benson Hill, one of our biggest investors, is Google Ventures. So organizations, companies that realize how important it is to better connect what farmers are doing and what consumers are wanting, and really helping to continue to improve our food supply. And what I see a lot of those kinds of companies doing that came out of this investment, they're very, very focused on consumer benefits. They're very focused on understanding the values of what consumers want, being transparent about what they're doing, but recognizing that empowering farmers with really great tools is one of the ways we're going to be able to meet that goal. And so there's just a lot more, I think, really productive dialogue around what a modern food system looks like and the role that science and innovation plays within that.
So transparency came up a couple of times. It's a word that is often discussed in food system discussions, and especially in gene editing. So I ask you to dig a little deeper on that. What is that going to look like in the future?
Okay. Well, I think that's part of what we're trying to really understand better as an industry today. There's a lot of work going on, focus groups, research with consumers, a respect that consumers want certain kinds of information, but at the same time, we don't want information to be misleading or so overwhelming that it just becomes confusing. So really trying to understand what is it that folks are most interested in knowing and what are the best ways for us to make that available. Some of that might be on a package, some of it might be through websites. A lot of what we're trying to do right now is just this kind of outreach that we're having. We had a session at South by Southwest a couple of years ago on gene editing. I would say that was pretty early days. We didn't have answers to all the questions, but kind of to your point, Catherine, I mean, we felt like we want to be out here talking about it. We want to get input of what people are thinking and what they're looking for. So I don't think that's all been completely worked out yet, Shauna, but I do think there's interest in that. We're happy that consumers are expressing so much more interest in how food is grown and what's in their food, and we need to continue to piece out what's the kind of transparency that makes the most sense for everybody.
Thanks. Anything to add?
I would say, I mean, coming from the farmer perspective, farmers are about 2% of the US population. And generally, they live in rural areas, and they might not be the most outgoing people all of the time. Not in a bad way, just a little introverted. I know that I am. So when I look at it, a lot of the time we haven't been telling our story. We haven't been telling people what we're doing on our farms, not because we feel badly about it or we want to hide anything, but because we don't come into contact with people all of the time where we would be able to do so. Or we don't know what people would think or if they would even be interested. And so working together with groups like NCGA, we've tried to raise awareness that people care about their food and they care very much about how it's being grown and what you're doing on your farm. And it's good to be able to talk about it, and it's good to get out there and push your boundaries a little bit and have an open discussion so that people can come back to when they knew farmers and when they knew what was going on in the production of their food, and we can build that bridge back there.
Thank you. So I have a follow-up question for you, Randy, regarding safety. So, you know, I think that topic comes up again a lot in the idea of responsible use and transparency. You touched on it a little bit. So tell us a little bit more about how, you know, what commitments you have in place, how your lab operates, what the governance is. How do you know that what you're doing is producing a safe animal for consumption in the end?
Well, I think the real question is what could be unsafe about removing a single protein, disrupting a single gene. I can't comprehend what it would be. If there's, I know we've kind of alluded to some off-site targets, and if that happens in a pig and you have an off-site target that's responsible for embryo development, you're not going to see the pig. And so we're not, in future generations, let's say there is a deleterious mutation that occurred, we will be selecting for the mutation we wanted, and those other mutations, those other edits will be selected against, and they'll disappear from the population. And so you have to also understand how the swine industry functions. It's kind of a pyramid system because companies have these nucleus herds where they'll breed in potentially this new edit, and then they will then feed down to multiplier herds, and then those animals will end up going out to producers, and those animals will grow those animals, or those people will grow those animals and then sell them, and that's really the animals that we eat.
Anything to add, build on that relative to plants?
Well, I think that a lot of what you just described applies to plants as well. I mean, there's the process that we've always been using with breeding we've been using for decades. And that too, there's a lot of mutations that happen, there's a lot of combinations that happen that we don't know what all of those are. With gene editing, it's a much more precise and intentional kind of change, and that's part of what allows us to make the exact kind of combination that we're looking for. And so I do think that it's a different story if we're intentionally changing, for instance, the nutritional profile of a plant beyond what could have been found in nature. So a lot of the edits that are being utilized or considered for the plant, for crops, we're really mirroring the kind of diversity that naturally exists within that plant species. We're just putting it into the right combination, but it already exists within that plant species. In some cases, we're actually going to change beyond what's in that plant, naturally existing in that plant species, And in those cases, there could be a different kind of oversight that's being done by the different agencies. So that's really the way it's being looked at, is depending on the kinds of changes that you're making and how that fits within the natural genetic diversity that's there, the different agencies, have different requirements around that.
Thank you. So I'm going to turn this question around a little bit. We've been talking about the risks of safety, off-target effects, and possibly even just perception of risk. What do you think the risks are of not adopting, of not having the ability to come forward with the solutions you're working on?
So I'll go ahead and I'll reiterate some things. We talked about, for CD163 and PERS, it's an economic issue, it's a sustainability issue, it's an animal welfare issue, it's a threat to our food security issue. But I think one thing that often gets overlooked is the psychological and emotional costs that farmers endure because we have not adopted these technologies. So when I go and talk to a producer group, and there's husband and wife sitting in the audience, and I talk about, well, what happens if a barn breaks and animals get sick? Well, the husband and wife often sit around the kitchen table at night, and you can imagine the stress between those two individuals when they're facing financial calamity. They're asking questions like, can we put groceries on the table next week? Can we send the kids to college? Are we going to go bankrupt? And so there's a whole psychological and emotional cost that I think people don't necessarily see when they see production agriculture. I'll describe this, and the husband and wife will be sitting there, and the husband, of course, he'll stare stoically ahead. The wife will tip her head to the side, kind of grab his shoulder and start to tear up. And I know that family has experienced exactly what I'm talking about. I think we have a moral obligation to adopt these technologies. The issues, again, yeah, economics is important. Animal welfare, sustainability, food security, the emotional cost that this disease inflicts upon families. Unless you have an agriculture background and you've been there, it's hard to understand. And I get it. A number of years ago, if any of you remember Paul Harvey, he was on the radio, and there was a call-in talk show down in Texas, and they were calling in talking about the farm situation and how the government's subsidizing farmers, and somebody called in and said, well, we don't need to subsidize them anymore, let them go bankrupt. And the person on the radio host said, well, where will people get their food? Well, Safeway. Don't they make it in the back? I mean, and so that's the mentality we're dealing with. People don't understand agriculture. And is that our fault or is that their fault? Probably both. But people don't understand. We have solutions. I think we have a moral obligation to pursue these solutions.
I would say, on top of that, if you look outside, when I walked in, I was chattering for like 20 minutes. I was so cold. Obviously, this is not normal November weather. And we're facing a lot of really extreme weather. And I'm certainly not a climatologist, but I know this summer, the drought that we faced in 2018 here in Missouri was very similar to the drought that we faced in 2012. In 2012, average Missouri farmer got 75 bushels per acre of corn. 2018, people worked on improving the varieties of corn that were available to farmers, the tools in their toolbox, so that the corn could keep growing even if it didn't have as much water. And we managed to actually still grow 142 bushels per acre. Now, in 2017, we had 170, so obviously it was a better year, and climate will still impact it, but by minimizing those impacts, you can actually preserve the crop, you can make sure that there's more food stability, and with a growing world population, we need abundant crops to make sure that everyone does have a nutritious diet that's plentiful enough to care for all the people around us.
And I guess I think of it as both macro and micro issues. So at the macro level, I don't know if folks realize this, but we need to grow more food in the next 30 or so years than we have in the last 10,000 years accumulated. And we're doing that in the face of changing weather. We're doing that in the face of dwindling natural resources. This is one of the biggest challenges of our time. I think I've heard Dr. Carrington talk about it as a grand challenge of our time, is how we're going to nourish all of this planet and take care of our planet at the same time. At the macro level, it's just really enormous. But I think about that as really a lot of that challenge is on the backs of my kids, all of our children, that generation and the next generation that are really going to be feeling a lot of that. And at the micro level, I think about how I want them to be able to, I want to use the best innovations, the best knowledge and science we have to improve the food that I'm able to feed my family every day in a way that aligns with the values of what I care about, which is largely around, their health and the nutrition that they're getting. And boy, it better taste great or they won't eat it. I know that. So really, I think of the excitement of being able to have so many other crops that we're bringing into the food system more than we have before. Things like plant-based proteins. Animal protein, I love animal protein. I like the choice of having more plant-based proteins from pulses and legumes. I like the idea of having better tasting greens that my kids are willing to eat and the best strawberries that you can imagine. I like the idea of being able to minimize pesticides by being able to utilize disease resistance that naturally occurs within that crop species. So I really feel like we're at a point that it's important for all of us to understand enough about how our food is grown, understand enough around innovation and science so that we're really trying to make informed decisions around risk-benefit. Because there are risks of not using these kind of tools as well, as we've talked about.
Thank you. So before we start taking questions from the audience, I want to bring it back home to Missouri. So Natalie, starting with you, how do you think this is important for the innovation ecosystem that St. Louis and Missouri is trying to build?
Sure. Well, I'm sure this is going to be touched on quite a bit in the next panel, but I will say, as one of the companies that's born of the innovation hub here, I've lived in Missouri now for about 25 years in St. Louis area. I think one of the most exciting things we have going on is the way that we have been able to build this innovation hub of using all these different kinds of science and technologies, the data science, artificial intelligence, but combining that with our real expertise around food and agriculture and plant biology that we here in the Heartland have. That's really exciting. And we've built, thanks to folks in the Danforth Center and many others, we've been able to build really all the things you need to make a company like ours, an early stage company like ours, really flourish. And that includes attracting investment from other parts of the country. It also includes attracting talent from other parts of the country into St. Louis and creating some really great jobs here. One of the things to realize about gene editing is, in our company, we're actually making these tools, we're using them in our own crops, but we're also empowering others to do that. Some of the innovators that we empower are companies that are doing things, like I said, directly trying to remove an allergen from food, trying to naturally decaffeinate coffee or something like that. But there's other startups here working, for instance, on microbials. Microbial is sort of like the biotics of like the gut flora that we have in our gut. It's what you really have for the plant. And they're really trying to optimize that so you can use less fertilizer, use less water, have more disease tolerance and things like that in the plant. Gene editing is one of the ways that we can optimize those microbes as well, kind of similar to some of the medical research that was done. So there's other startups here working on cover crops. These are crops that are really important in terms of soil conservation, have had almost no genomic innovation. Through gene editing, we're able to make a lot of the improvements much more faster than we could have through traditional breeding. So I feel like this technology, it's not just about the startups that are actually making those tools or using them directly, but about how we are actually helping other innovators working in other ways of improving food and agriculture do it more precisely and quicker than they could have otherwise.
That's great. So, Dr. Prather, tell us a little bit about it, the university research community and how they're thinking about the opportunities.
So the opportunities are grand. There's incredible opportunity. One of the things I didn't talk about is that I'm also director of the National Swine Resource and Research Center at the University of Missouri. We're a core facility for making mutations in pigs or edits in pigs that mimic human disease. And so we've made about 70 different modifications in pigs already to create models of human disease. To have those technologies available to us allow us then to do things in agriculture, allow us to do things in medicine, and I think it really builds off one another and helps address the One Health issue. We have tremendous strength here in Missouri with this technology, and the applications are really wherever your imagination can take you.
So Catherine, you get the last word. What does this mean for growers in the region?
Sure. Obviously, for the farm families that rely upon having a crop come in every year just so that they can plant the crop the next year, this is their livelihood, and it's what they want to give to their children as their farm, and the good land and the good water and the good air that will allow them to continue the way of life that they love so much. From a more macro level, Missouri is the 9th largest corn-producing state in the country. That's $1.1 billion every year that corn is putting into Missouri's economy in terms of a benefit, and 22,000 roughly jobs it is directly supporting. On top of that, having that much corn and investment in things like ethanol and other clean fuels allows more jobs even in Missouri. And when you look at exports on the Mississippi River, again, the economic benefit just grows and grows. So it's good for the farm families, but it's also good for all of Missouri. And being able to keep growing the crop to deal with the challenges that we're going to be facing in the future and that we are right now in a more efficient, better way is going to be a great thing for the state and for us.
Fabulous, thank you. Well, we have a number of really great questions here, so if there's others in the room, I think we'll have time to take a few more. So the first one is for Dr. Prather. Could you talk a little bit about how CRISPR might offer potential to breed pigs for organ transplants to humans?
So that's one of the emphasis that we have at the National Swine Resource and Research Center is to create pig organs that can go into humans. We have some pig organs that have gone down to Emory, for example, kidneys, and they've been life-supporting in primates for about 2 years now. And so that's very encouraging. The CRISPR-Cas9 system just allows that editing to happen so much more efficiently. When I first started working on these projects to knock out genes, somebody would come to me and say, how long is it going to take? And I would say, well, it might take a couple of years because we have to build a construct and we have to do homologous recombination, and then it may still not work. So I have one example of a project that actually took about 10 years before we knocked the gene out. And then the CRISPR technology became available, and within 6 months we could have the pigs. It just so totally changed the landscape about how we can move forward. And so yes, we are working on organ transplantation. And again, that helps us to go in and deal with those molecules that affect rejection. So when transferring a pig organ into a human, the first molecule that affects rejection is a molecule, it's a galactose alpha-1,3 galactose sugar linkage. It's a molecule that sticks up on the cell surface. Everybody in here currently has coursing through their veins right now antibodies that recognize that molecule. The molecule's on the surface of bacteria, and you've been exposed to bacteria, so you have pre-existing antibodies. And if we transferred a pig organ or cell into any one of us, you get hyperacute rejection. Within minutes, the cell, tissue, organ, whatever, is destroyed. In 2002, we went in and knocked out that gene, the enzyme that puts that molecule on the cell surface so it's not there. So now those pre-existing antibodies don't have anything to recognize. Well, what happened then is we started seeing all the other things that crop up in terms of an acquired immune response. And so we're slowly dealing with those and trying to address those. So yeah, this technology, not only does it have benefit for agriculture, but it also has tremendous benefit for biomedicine.
That's fabulous. So we had one question about GMOs, and that's actually something we didn't delve into in a lot of detail. So maybe, Natalie, could you talk a little bit about how, first of all, how gene editing and GMOs relate or don't, and then what the evolution of these tools might mean for GMOs in the future?
Sure. So one way to maybe think about it in a simplified kind of visual in your mind would be if you are trying to get your ultimate outcome is a large red apple. You want the size and you want the color of that apple. And through traditional breeding, we would take a large brown apple and a small red apple, and we would be breeding them and breeding them and breeding them, trying to get to the combination of both large and red, and that takes a long time. With GMOs, what you might have done is you would have taken, let's say, obviously this is a simplified example, but you would have taken the gene for large size in a pear and put it into your small red apple, so that now you've got large size with your red apple. But you've actually taken a piece of DNA from one species and put it into another. With gene editing, what we're able to do is go back to that large brown apple, find the particular gene for size, and go to our small red apple and make the change within its genome so that size is upregulated. So now you have a large red apple again. So gene editing can be used to make GMOs if you wanted to do that, but the way we're generally talking about it within the industry is it's about making those very precise intentional changes within the genome of the same species. So you're not taking DNA from one species into another in your final product. Does that sort of explain it for folks?
So thanks for that explanation. I'm going to just interpret some meaning here. I think people might want to know what does this mean for GMOs? Will they still continue to be developed? How does gene editing maybe change the path or not for GMO development in the future?
Yeah, well, there's certainly have been a lot of benefits of the GMO products that exist today, and I expect that those will continue. I think a lot of the companies that have traditionally developed GMOs, as well as many other companies that have not done as much work on genomic innovation, are really focused on gene editing because of that precision and the time improvements and things like that. One of the things that's really going to be important as we think about the regulatory environment for that is that we not create, because these are very different in terms of a risk profile when you're not crossing DNA, we really want to develop a regulatory system that's not going to be creating a barrier to entry for gene editing for its application in smaller crops and by smaller companies. But I see there's going to be a lot of interest in gene editing across the board. However, there's also going to be instances where the kinds of changes we're looking for do not exist within the natural genetic diversity within a certain crop. And you're really going to need to be able to utilize DNA from one species into another. I've heard Dr. Raven, who's used to be one of the premier conservationists, who used to be with the Botanical Garden, talk about that and the importance. That's one of the things where I think GMOs will still have a role in the future. But I think you'll see a lot more focus on gene editing within a crop species for our food system.
So several people in the audience are curious about the European Court of Justice opinion. And just for context, for those who might not be aware, very recently, earlier this year, Europe asked a question of their system, and it went through a legal route to understand how gene editing would be interpreted. And that European Court of Justice ruling was that gene editing was considered a tool of mutagenesis, and that all tools of mutagenesis fall under their GMO directive. And so what that means, and I think a lot of people are trying to understand this legal ruling better, but the preliminary read is that something created through gene editing would have to go through the intense kind of regulation and studies that a GMO does. So to the panel, provide your perspectives on that for the group. What do you think that means for U.S. R&D as well as adoption of crops and animals in the long run?
Sure, I'll start. It's interesting that we have different rules at different places. So we could take a pig and use ENU-mediated mutagenesis and introduce hundreds or thousands of random genetic mutations, and we can eat those animals, and there's no regulation of those animals right now. But because we use the CRISPR-Cas9 system and we go in and we precisely edit 1 or 2 letters of the genome, now it has to go through FDA approval. So I'm not sure how ENU-mediated mutagenesis is handled in Europe right now, but what you implied is that's regulated. But how would you know? How would you know somebody did it?
I would say, while the European ruling does add a new sort of look at the regulatory system from an international level, I don't think that it's necessarily any different than what we've gone through with GMOs and the incredible difference of regulation between our export markets that we've seen under GMOs, with incredibly slow, if any, approvals in a place like Europe, and an asynchronous and at times non-functional regulatory system in China that lacks transparency completely. And then you have countries that have a functional, timely, science-based regulatory system like Japan and Mexico and Canada. And I think that farmers have to really understand where their crops are going and take some responsibility for that. But I know in some crops, it's been more important to preserve a market and not have a tool. In corn, we really want access to markets, but we want them to be science-based and functional in the regulatory system. And we also want access to the tools we need to meet tomorrow's challenges. And so there has been at times a look at, is a market actually really functional, and is it even going to buy corn from us in the first place? And at times we've decided to steward corn but launch traits that have not been approved necessarily because we know it's not going to go there. We're going to keep it in a stream and send it to where it is approved. And that's been our approach. We've adopted technology because we need it. We want markets, but regulation has to be somewhat predictable, and there has to be a scientific basis to it, or else we're being deprived of the tools we need to do our job for no reason than someone's emotional or politically based process.
I think it's important to recognize that most of the academic community, scientific community, really sees this kind of tool as being quite different in terms of that risk-benefit analysis. And we're seeing in a lot of governments around the world, the United States, Japan, number of parts of Latin America, really what I would consider to be a very risk-based, risk-benefit kind of approach, scientific kind of approach to how they're managing oversight, different from what we're seeing with the EU. One of the things I think was really interesting is Center for Science in the Public Interest, which is a consumer watchdog group. Their primary focus is around a more healthy, safe food system for consumers. That's their watch out. That's their mandate, their mission. And they were one of the first to write a blog about the EU ruling, basically expressing that this isn't really a risk-based kind of ruling in the way that they've done it. It was really about how much time you've been on the market. But it kind of goes to Dr. Prather's point of here we're making very intentional, we understand the kind of change we're making. It's a very different kind of approach and should be considered that way as we look at oversight. One of the things I think is really important to understand is if we're going to use the same kind of approach as we did with GMOs, where you're actually moving DNA from one species to another, if we're going to treat the kinds of changes that we're making within a plant that we can be making through traditional breeding, if we had, it's really just a difference in how much time we're taking. If we're going to treat that like we are a GMO, you're essentially going to recreate the model where just a few of the largest companies with the largest budgets focused on the largest crops and a few kind of benefits are going to be able to make genomic innovations using this kind of tool. If you really want to be able to enable smaller companies, companies working on lots of diverse crops, some of which are smaller, companies focused on consumer benefits, to be able to use this kind of tool, we really need to make it very science-based and appropriate for the kind of risk benefit that that specific product provides.
So if I can jump back in, I need to make a distinction between plants and animals, because plants don't get regulated by the Food and Drug Administration, Again, we make one letter change in the genome and the FDA regulates it as a new drug. Genus, who we've licensed the technology to for the PRRS-resistant pigs, estimates this is going to cost $50 to $60 million to get approved. And we talk about trying to reduce the bar so other companies can get in and participate. That isn't conducive. I think in transparency, we also need to be a little bit careful about what we're talking about, because we talk about gene editing, and I'm talking about changing a couple letters of the genome. We also talk about GMO, and that's maybe bringing in another gene from another species. So for example, we've brought in a gene from C. elegans that creates, or that takes omega-6 fatty acids and converts them to omega-3 fatty acids. So we have pigs making their own omega-3s. That's clearly a GMO. But what about something in the middle? There's a group in Roslyn that's very concerned about African swine fever. And what's interesting is the warthog gets African swine fever, but doesn't get sick from it. And so they're looking at, well, what's different between the domestic pig and the warthog? And there's a protein called REL-A that has 3 amino acids, 3 letters of protein that are different. And so they've used a gene editing technology to introduce a change in those 3 amino acids. So is that gene editing, or is that now a transgene? And so we, in transparency, we do have this continuum, and we need to talk about it so we understand it.
Good point. So, the last couple of questions here actually are right in line with our theme, so I'm going to kind of combine them a little bit. And so talking both about nutrition and sustainability. And so the questions are around specific, how can gene editing help with specific challenges, for example, drought, and in the case of sustainability, and in the case of nutrition, making healthy fruits and vegetables more affordable and a higher mix of production. So any thoughts on what you've seen folks doing, or what the potential is there?
So when I look at sustainability, what we want to do is create pigs that don't need any inputs. I want pigs that we don't have to vaccinate. I want pigs that we don't have to balance a ration to, that we can feed them low-quality feedstuffs and they'll still perform. I want pigs that have meat quality that is healthy for us. We can use these technologies to do all of those things. And if we're really concerned about sustainability, I think we need to be doing them.
Yeah, I'll just add, so for instance, I mentioned plant-based proteins earlier. One of the challenges there can be having the right kind of texture of those so that you can actually make them into the kind of products that we're used to eating. Another example would be sometimes they don't have as complete an amino acid profile as animal protein. So what can we do to try to improve their amino acid profile? We can work to remove anti-nutrients from certain crops. I talked already about removing bitterness from some really already nutrient-dense crops. So there's lots of ways that we can be thinking about improving nutrition in the food system by making more nutrient-dense crops usable in different ways, or by actually improving the nutritional profile of crops that we're already utilizing a lot, as examples.
So I want to thank the panel and for the audience for great questions. I think it's, as promised, I think you heard excitement. I think you heard about benefits. I think you heard about how we want to really, engage with the consumer in this, and how important this technology is for this region and for the future. So thank you very much.
Hello? Oh, okay. So thank you so much. What a great discussion. That was amazing. Our third panel is gene editing to create 21st century jobs. And I'd like to invite the moderator to come down, Vijay Chauhan. He is the global STL lead for BioSTL. Okay.
Thank you, Dina. Good morning, everybody. It's time to wake up and have another great conversation. I'd like to have my colleagues join me on this. Chris Ote from Alexandria, Supriya from Millipore Sigma, and Sam Fiorello from none other than the great Danforth Plant Sciences Center and Bridge Park. So, as you all heard, the topic is going to be about gene editing and the 21st century jobs that it will create. But before we start the conversation, I'd like to get a feel for where the audience is on a couple of questions. But let's start with one. How many in the room here, now having gone through what you heard this morning, what you knew before you came here, think gene editing is going to be one of those big transformational technologies that's going to be key to the success of not only humanity, but also some of the industries in the world? Raise your hand. Okay. How many feel it's not going to be? Are you scared? Or are you convinced? Just trying to get a feel for where we are. That way we know how we can interact with you to have this conversation. And I'm really amazed that you all have raised your hand given all the discussion that took place and a lot of question marks about, is this thing going to go down the path of GMOs, or is this going to will have its own new path and be able to deliver on the full promise of what this technology represents? So we'll take where you are as face value. So I wanted to do a few framing comments for this conversation that we're going to have. As you probably heard, gene editing is the latest innovation in the field of genetic engineering, where one big milestone started with Gregor Mendel in the 1850s, when he established the laws of inheritance. And then we lurched forward. In 1978, the Nobel Prize for Medicine was given for the development of rDNA technology, recombinant DNA technology. And so gene editing is sort of the next path there. And just like rDNA technology really transformed big industries, from healthcare with the production of insulin, which prior to that was derived from animal sources and now synthetically produced, and a whole series of biomedicine products, and in agriculture with the development of BT traits and Roundup Ready traits that again transformed agriculture. Similarly, gene editing is going to have that kind of dramatic impact. And since we're talking about the economy and the 21st century jobs, what's really important to know is that St. Louis was ground zero for that technology and those industries. And really, what's really interesting is this panel that we have. It would have been really hard to see a panel like this take place in Boston, in California, because while they have one industry, they don't have the other. Here in St. Louis, we actually have powerhouse industries in healthcare, human healthcare, and in agriculture. So that's what makes this a really exciting place to have this conversation. So what we're going to be talking about in this conversation is a little bit about how gene editing is going to transform these industries. Sounds like we covered a lot of that in the previous panels, so maybe we won't talk much about that unless you have a compelling example or two. What we want to talk about is how St. Louis is poised to be a global hub in gene editing technology, gene editing industries, and the 21st century products, markets, economy, and jobs that are going to be created from this innovation capability. So that's what we're going to talk about. Is that okay with you all? Yeah? And then through your questions, if there's anything missing, put it out there. This is a conversation for you, with you. So if you're not getting what you want out of it, you got to raise your hand. And we are here just going to do our best to answer. So luckily for me, I have a great panel to help me out. And so I'm going to start by having, so I've got Chris Otte from Alexandria Ventures, going to provide a fantastic perspective, the voice of the investor, the voice of an organization that has built innovation ecosystems all around the country. We have Supriya from Millipore Sigma, really accomplished scientific leader and speaking on behalf of the human health side, and then Sam Fiorello carrying a huge burden of responsibility for our ag food tech ecosystem, for which he has been an instrumental part in building. Great panel here, and I'm really excited. I'm going to let them introduce themselves. So in a couple of minutes, Chris, tell us a little bit about your background and how you come to the gene editing space.
Sure. So hello, everyone, and thanks to the organizers for putting this together, this good event. I think Vijay said it pretty well. So I'm actually from Boston. We try to put together these events. We don't have the ag kind of infrastructure that you guys have here, too. So that's a really, really unique element. So I'm a scientist by training, did a biochemistry undergrad, PhD, finance and MBA, worked in tech transfer a while at MIT, and then joined this real estate company, Alexandria, about 8 years ago. And Alexandria has kind of two unique aspects to it. One, we're a publicly traded real estate investment trust, and as Vijay said, we look for markets in which there's good science, good talent, good capital, and good infrastructure, and then we kind of go in and help to facilitate the growth of that ecosystem as well. And so Boston's our biggest market. We're mostly in the biopharmaceutical space, which is why we don't have really a presence here in St. Louis, although we do have an investment, which I'll talk about. San Francisco, Seattle, San Diego, North Carolina, Maryland, and Manhattan. So those are kind of markets we're in. The company's about 25 years old, and for about 22 of those years, we've also had an on-balance sheet venture capital fund. Today, it's almost $1 billion of assets under management, and we've used it to invest, to learn, and also, hopefully make money as investors. And what we really look for is cutting-edge new areas of biology, chemistry, and new, really groundbreaking platform technologies that we think will be very impactful to the future of humanity. And also create really strong companies. And what's really kind of different about us is we have a very long-term perspective. This isn't our core business. It's very strategic. It's very relationship business. So, we were early involved with a company called Alnylam, which is one of the first companies in the RNAi space, which you guys have probably heard of. They were founded about 20 years ago, and they got their first product approved in August. So it takes a while. Gene editing has kind of been through some similar things, and we find it interesting because it does follow that too. And what I think is unique about it, and I think some of the previous panels said this well, I think Dr. Prather actually said it really well. It said, we could always ask these questions of like, oh, if we could, genetically engineer a pig for, that purpose, or one I'll talk about as well, or if we could engineer human cells for a therapeutic modality, wouldn't that be great? But you couldn't do it because you didn't have that technology. It would take 10, 12, 15 years, and it would cost way too much. Gene editing allows us to do all sorts of new stuff we couldn't do today. That makes it a particularly good investment opportunity for us. I'll get into that later, but
Yep.
So that's me.
Great. Thank you, Chris. So Priya, can you tell us a little bit about yourself and how you come to the gene editing space?
Sure. So I started off as an academic and followed that up with a postdoc, a PhD and a postdoc, and then went to a number of small startups in the Bay Area and realized that I had a love of just understanding what was going on in the world in the big picture piece. And so got completely seduced by the dark side, which is the business side of it, and understanding how business can drive science. Like I think you just said too, is that investment is a big part of how things move in science, and we see that with the CRISPR wave. But I ended up joining Millipore Sigma, which is now pretty much the opposite of a startup in the sense that it is a giant company. It's known as Merck overseas. And we realized that Sigma, which was one of the companies, and Millipore, which is another company, and what we do is we make products for pharmaceutical companies, for plant companies, for pretty much most industries, flavor and fragrance companies. We make a lot of the products and we work with them, and we realized that we had a deep understanding of gene editing, and I'm going to come back to that in a second. We had a deep understanding of that, but we also had a very strong play into manufacturing for products for human health, so for drug products as well as gene therapies. And when I say I wanted to come back to the idea of ground zero, is Sigma was one of the very first companies to ever offer a gene editing solution, which was called zinc fingers. So St. Louis, where Sigma had been for 80 years, I think they celebrated their 80th about 3 or 4 years ago, was truly ground zero. It was the first commercial. We had the technology from another company in the Bay Area, but it was the first commercial use of gene editing, where we actually developed this for the research community to use in their labs. And with that came the education piece. So there are so many people in St. Louis that have been working with gene editing for 7 years before CRISPR even came about and was used. So St. Louis truly is a ground zero for this technology. And we actually, because of that, we know so many people in the WashU who have taken this technology much further, the startups that have taken it, Pfizer, Monsanto, everybody who has taken the gene editing technology so much further in their applications of what is essentially a tool, right? Gene editing is a tool. It's the applications that are bounded by the limits of your imagination. So having had a head start in this, I'm learning what it's like to be in a startup environment in a 50,000 person company, which luckily has access, and we work with our customers in plants as well as in human health and all the other cosmetics industries, all these other places where you can see all these applications for gene editing.
Sam?
Yeah, hi everyone. I'm Sam Fiorello. I work for a 300-person startup called the Danforth Plant Science Center. We just celebrated our 20th anniversary on Saturday, actually, and I wear several hats and I think several points of connection to this subject here. One is here working at the center with Dr. Carrington and his team of scientists. I am also the president of the research park, the Bridge Park on campus, and our mission there is to try to serve as a catalyst to help move technologies from places like the Danforth Center into the world through products and services and companies. Interact with the Helix Incubator right across our parking lot, and also been involved in this 39 North initiative trying to create a larger innovation ecosystem, live, work, play kind of a thing, kind of stuff that Alexandria is really good at and has done all over the country. I am bringing the perspective of a non-scientist, a lay person, so I can say all the dumb things and it's okay. So I'll leave it at that.
Yeah, great, Sam. Thank you so much. So Sam and I were just talking. We are really feeling the pressure to make this a great conversation for you all. Okay, so really going to try and put out some things there that will be very helpful. So Chris, I'm going to flip around our plan and go to you on one thing. You guys have been building innovation clusters around the country. Can you tell us, with your experience, what are the key elements for success to be successful in a particular industry? What do you have to have in place? So the audience can start to get a feel, and then Supriya and Sam will talk about what do we have here against that framework.
Yeah, so the way we look at it is kind of, there's 4 pillars. It's kind of innovation, capital, talent, and, infrastructure really are the kind of the 4 elements we look for. And nowhere's perfect, right? And I will say I'd say this to every region, we do a lot of, we meet with European companies and other groups, and they're like, how do we be the next Cambridge? And I'm like, don't compare yourself to Cambridge, don't compare yourself to the Bay Area. Those have been informing for 20, 30, 40 years, and, the timing was impeccable. So that's always a bad baseline. So then you start to look at other markets like San Diego, Seattle, North Carolina, and I would then start to say St. Louis has most of those elements already. I mean, there's really, really good talent out of WashU, Danforth, other local universities. The Bayer and Monsanto kind of organization being here so long has created a lot of very talented people. There's good infrastructure. We're investors in Benson Health, so we know that they've relied heavily on the Danforth Center and the resources there. It would have been hard to kind of do what they've done in really any other location, I think. Science is great too. As you heard the 2 folks speak earlier, great science here as well, also on kind of the cutting edge. I know there's a lot of cell therapy work going on at WashU, a lot of human health going on as well. And then, but ultimately I think it comes down to the people and like I said, the talent. And so you have a lot of, I think, really young, smart people coming to universities. You have people at Bayer and Monsanto. I think what we probably could use more of, and I think we definitely know from Benson Hill and a few other companies I know in the area, is having people go from the big companies into the small and back and forth. And I'm sure that's happened here. But if you look, especially in Boston, that's what kind of one of the magic things is, this really powerful informal network. And people, really senior management, if you talk to most, at least biotech CEOs, if somebody comes to them looking for a person to hire and they have someone really good in the organization, they're not going to fight it. Because it's more about the mission of the location and impacting human health and impacting humanity. And if someone being in another company is a better way to do that, that happens. And then we have enough companies there that, you know, if one of your companies doesn't work out, you can go to another one. We have like 500 biotech companies. So I would hope that we see that, you know, here and in a few other markets. We have a big presence in North Carolina as well, that we start to see that in agriculture, you know, and I think this gene editing wave will help to push that.
Great, thank you, Chris. So I'm going to put up a graphic here that is a visualization of the St. Louis life sciences ecosystem. And if you just look at that and you look at what Chris just shared with us, you'll see a great mapping between the elements of an ecosystem that they have seen in other places, and we have one here in St. Louis. And what is ironic is for most of us, we are either unaware of it or we take for granted what we have. So I'm going to put a huge burden on Supriya to make a passionate case for the great ecosystem we have here in life sciences. And then Sam will have another slide, and we'll talk about the ag and food ecosystem. So hopefully through that, you'll get a sense of the scale and the depth that we have here in St. Louis that are going to be critical for success in the gene editing space, because it's just a progression on a path that has already been laid here in St. Louis. So Supriya, tell us a little bit about what's going on at the back here.
Okay. I'm going to take one quick small step back just to highlight gene editing of, you know, why it plays into this kind of spiderweb network. So gene editing itself is the tool, as I said, but it's the applications are myriad. And in the discovery, in the medical space, and you heard our two doctors talk this morning, scientists talk this morning about how they use it in their discovery work. So if you split it up into 3 areas, you can use it in discovery, so you understand and you can build better biological models to understand disease. You can use it in preclinical safety and saying, how can we make this a more targeted approach for human health, and how can we make sure that there are no side effects from it? You can build models to assay that. You can look for immune responses, as was mentioned earlier, and make better tests for that using gene editing. And then you can use it in that manufacturing side for gene editing, for manufacturing products, whether they're small molecules like drug compounds or large molecule ones like antibody ones. And what you can see is when you look at this large network is that we have players that play in all of these different areas, including some of the side ones, like was mentioned by Benson Hill, which we actually have a collaboration with on the microbial side, because we can use CRISPR gene editing in microbes as well to actually improve human health. And what you can see is about 5 years ago, there was probably three major big companies that were supplying different gene editing tools, and the advent of CRISPR, which was shown how it works, et cetera, it became a very affordable, easy to use tool for everybody to be able to play. So in 5 years, it's gone into about 80% of the research labs worldwide, and is now used almost like PCR is. So what really stands out is not necessarily that everybody here is using CRISPR, but it's that they all have different applications that they're using CRISPR in, which means that anybody who's got a really, really good idea, this is a great place to take that idea, and if you can apply gene editing to make that idea come to fruition faster, which is really what this tool is about, then you get startups. And what you can see is we have the bigger players that have been in the field for a long time with the Pfizers and Millipore-Sigma and Mallinckrodt, et cetera. But the number of startups in the bio-STL area has just grown, and they've all come up in the last 4 to 5 years to more prominence in this area. And they all are doing something slightly different with CRISPR. So what that means is everybody is adding synergistically to the value and the power of CRISPR and the ability to trade new ideas about it in the St. Louis area. So that's also attracted our investors. And it goes all the way down to not just do we have startups and incubators, but we also have contract research labs that are really involved in the manufacturing process as well of this, of really getting this from the bench all the way to the bedside. And that is really can't be done without research partners like WashU and other universities that are playing into this.
Great, Supriya, thank you. What you all may not know, as Supriya was talking about, is we have a very vibrant startup ecosystem in the life sciences area. It's probably one of the best places to start a company because there are so many resources to help you get a company off the ground. Our organization, the Biogenerator, is in the middle of that. And our company is now raising large amounts of capital, which was not possible many years ago. We just had a company, Galera Biosciences, that closed a $150 million C round. We had Benson Hill right here, closed a $60 million round in the ag sciences. As Natalie was saying, it was the first investment by Google Ventures in the space, in the ag space. This is the kind of attention and understanding people in a very sophisticated way have developed about St. Louis. So great stuff going on in human health sciences. Sam, I'm going to have you take us through a little bit about what's going on the ag and food side.
Yeah, so I think in order to set the table and to bring it together, I just came back. I spent 2 days, the last 2 days, on a farm in New Madrid, Missouri, talking to the farmers there, and there's frankly their struggles trying to break even or make a little bit of money in a world where there's $7 and change soybeans and corn. And I work here in St. Louis in this innovation ecosystem. And when I was driving back last night, I thought, you know what, with a new technology, when a new technology like gene editing, CRISPR comes along, it has the ability to really benefit both rural America and innovation centers like this. If you look at the food chain, the production system, and you look at $1 of value, today especially those farmers that I was with, about 10 cents of that dollar goes to them. We also heard that the average farmers in their late 50s, and less folks are saying that farmer, part of it is the economic reality. If you're a young farmer trying to get into this and you have to take on leverage, in this kind of commodity price, it's very difficult. And so it's painful in rural America. But a technology like this comes along, it brings the cost down of creating solutions that those solutions then could change the paradigm in the food system so that the farmer can capture 15 or 20 cents of that dollar. And here's why. The Benson Hills can now create crops and put value in that corn or soybean or sorghum crop and contract directly with Tyson or Pepsi and say, hey, we'll go straight to you because the Benson Hills using this tool have brought the cost down to create it so that they can customize a product where that product can go straight to the chicken feeds or turkey or whatever Pepsi wants. I believe that will really transform rural America and be a real boost. It'll also sort of blow up our commodity system. So there's opportunities and challenges for Chris and his folks and all the innovators out there. For St. Louis and this innovation ecosystem, what it does is, and we've seen this we have an annual investor conference called the Ag Innovation Showcase, and I think it was about 6 years ago, we have 4 categories of attendees. We have investors, we have entrepreneurs, we have key strategics, and then thought leaders. And the thought leaders are there to push us beyond 6 months and 1 year. And maybe 5 or 6 years ago, someone said, this new technology, CRISPR-Cas9, is going to change the way we all work here. In 5, 6 short years, the 2018 Ag Showcase, half or more of the companies presenting, a vital tool for their business was gene editing and CRISPR-Cas9 and the other CRISPR technologies. So that is now leading to more innovation, more startups. Dr. Levine said that he now has high school students using this tool to make advances. So it's faster and cheaper, and it's this great equalizer, because it doesn't have to just be a company the size of a Monsanto or Bayer to go after these things, because it's so expensive and there are huge hurdles. It can now be a company, a startup like Benson Hill entering this. So I think it changes so many ways. To point back to this slide, when something like that comes along, how does a community take advantage of it? You have to have the pieces in place that Chris talked about. You have to have places of discovery, like the Danforth Center in WashU and Mizzou. You have to have risky capital. You have to have specialized facilities like we offer here at the Danforth Center, greenhouses and growth rooms. You have to have a talented workforce, a workforce that can take on risk and launch these things. You have to have access to markets, the farmers and the food processors and all the rest. We have all that here, and in many of those bubbles, we're best in class in the entire world. So it's an exciting time for this and this confluence of this great new technology, and I think we're poised to take advantage of it.
Great, Sam. Two excellent points. One, the democratization, if I can pronounce that word well, of what this technology is doing for innovation. One big point there. I'll have you talk about that in a second, Chris. And the second one is how we are poised here because of what has been built over a period of time to really take it to the next level. Imagine if you wound the clock back 20 years, if you looked at this chart, you pretty much would have that first bubble, corporate strategic partners, and a little bit on the research part. Danforth Plant Sciences didn't exist, and we didn't have the investors, we didn't have the innovation communities. Those are the missing pieces. If you want to create a startup culture, as Chris articulated, you got to have all those other pieces. Danforth Center came along, the first 10 years, they put their heads down, building high quality innovation. And then Sam and Jim come along and said, we're going to blow this thing open and create an ecosystem where startups can move fluidly between the assets of the Danforth Plant Science Center. The scientists here will work with the startups there. All of that is the substrate on which we're going to be now building this gene editing innovation platform. Doesn't happen overnight. You just don't flip the switch. It takes 15 to 16 years. That's a huge asset. That's what Sam was talking about. In terms of this democratization part of it, Chris, speak to what this actually represents as an innovation opportunity, as Sam was just talking about. What was, what's the sort of big structure today through which innovation was going through life sciences, and how it just blows open the scale of innovation that's possible because anybody can play in that game.
Yeah, we're not quite there yet, but we're getting closer. So I'll maybe kind of step back and talk about, so we've been investing in the gene editing space for, I guess this wave of gene editing space for 5-6 years right now, kind of before it became so hot. And I think, I then take it for granted and forget that stuff, like the stat that he was shared. So I'll just give you a few examples of companies we've invested in that talk about using this tool, as was alluded to develop an actual product, because that's what you need to do. I mean, it's a great research tool. It's powerfully used by academics. They get great results. But ultimately, I think we all, academics, investors, and business people want to see a product get to market and then generate revenue, which is what kind of generates these jobs and generates this virtuous circle in these economies we're in. We hope to see that here in kind of the St. Louis area. A few examples. So about 5 years ago, we invested in a company that's now named KSQ Therapeutics. They're in the human health space, and they basically raised a little bit of money and said, hey, we're going to go use CRISPR and just screen the whole human genome against, cancer, see if these genes are cancerous or not. It's like, well, what are you going to patent? It's like, we don't know. We're just going to do it faster, cheaper, and we're just going to do it first. So they did it. That was about 3 or 4 years ago. They've uncovered, so you probably heard of like these immunotherapies, PD-1, PD-L1 inhibitors. they have these amazing charts that show, you know, the PD-1s and the known IO targets, and they have ones that are like tenfold better that they've already discovered using this platform because they're non-obvious. Now they have compositions to these targets. So they kind of said, here's what we want to do. Here's the crazy idea we have. We don't know how we're going to form a business around this, and they did it. You know, and they raised $75 million, and Genzyme's former CEO, David Meeker, is now leading the company. It's a Boston company. So that's a really cool example. In the pig front, I'm sure you know eGenesis in Boston, so we're invested there. They're using CRISPR to remove the antigens, you know, the pig retroviruses, to then ideally have a universal pig donor for human organs. That'd be great. You know, will they solve the whole organ transplant problem? Well, partially, who knows? But any of that would be super beneficial and very valuable. As I mentioned, Benson Hill, it's probably a special example for us. We were investors early in the A. One of the companies that's not corely located in one of our regional markets. We've been big supporters. But again, democratizing these gene editing tools to, I think, what will enable what I think is really exciting is, you know, if you look at the life sciences and healthcare, what I kind of think is ag right now lags about 5 to 8 years behind in cycles and trends. And so in pharma, why Kendall Square is so popular right now is this externalization of research concept. So pharma is scared they're going to miss out on the next innovation, wants to be as close as possible to the startups, and is working with them in every way they can. And that's really enabled companies in that market to raise that money. And a lot of these companies can raise money to go after very precise diseases. So the concept of precision medicine, you know, that kind of really took root in oncology because we understand that best, but the cardiovascular stuff is exciting, fibrosis, kidney disease, and obviously the rare diseases. And what I hope we see is in agriculture, we see the same thing. So you can imagine a statewide or regional-wide specific crop, you know, whether it's corn, whether it's soy, whether it's whatever, that you can grow locally, you can grow, you know, in a very specific way with good nutrients, you know, good benefits. Ideally remove the shipping costs, remove a lot of supply chain, as alluded to. And so that value goes back to the farmer. So this precision ag thing and external research, I think, will be a big driver for agriculture in this market too.
Great, excellent examples of what this democratization process looks like. So Priya, do you want to share with us sort of how this whole process of discovery, development, and bringing products to market is going to get transformed through gene editing, and the kind of jobs that are going to be created and the kind of jobs that are going to kind of either disappear or become less important? I want to help the audience get a feel for the job skill set landscape, how that's going to get transformed. Because it's not going to happen and everything's going to, you know, this kind of transformation is not possible and everything else is going to remain the same. And for the audience that's looking to build their careers over the next 30, 40 years, it'll be good to lay out a landscape of what's coming at them.
Sure. So for the first part, sort of how gene editing is transforming, I think we've heard a lot about how it's the tools that are really going to change the applications. And I'm going to give you one example that Millipore Sigma worked on. So if you're going to, how many of you, I guess, have ever taken any kind of a drug, even over the counter, and it has a little black box warning, or it has a little thing that says, and the ads that say, may induce nausea, you know, vomiting, et cetera, et cetera, et cetera. And that really comes back to that point that was made much earlier about medicine in general. You treat everybody the same. Personalized medicine is this new approach to really trying to understand how you are different, and if your genetics play a part in that, how we can use that to actually change the treatment. So one of the ways that gene editing is completely transforming this whole space is, by example, I'm going to use the CAR T example, where what is actually happening is they're taking your own cells so that you don't induce strong immune responses. You take your white blood cells, so they take the blood out of you if you have a leukemia or a blood-based disease. They take the blood out of you, they take the white blood cells out of you, they take those T cells, they engineer the T cells so that they are now able to recognize the cancer antigens or different proteins that are on the surface of the cancer cells. So they engineer those T cells. They're your own. They grow them up in large quantities. Then they take those and they put them back into you. Though now these re-engineered T cells in you are basically going after the cancer in your blood. How this is changing it, though, is that this is not a treatment, it's a cure, right? You're not coming back over and over for more different types of treatments. You're not taking a pill for the rest of your life. This is a cure. And one of the ways that gene editing is completely uprooting this whole system is it's really saying, we don't really deal with cures and medical treatments in general. We keep seeing patients year after year, and you keep trying to treat the symptoms, and occasionally you can treat the actual disease. So how do you pay for something like that? So what we've got is by gene editing, developing a new technology that actually changes how we treat disease, you're actually creating this entire ecosystem of people who are now going to have jobs that are going to be related to very specific aspects that you didn't expect. Because if you expected 5, 10 years ago that somebody in reimbursement and pay accountables, et cetera, and finance is going to have to actually be paying a lot more attention, well, that day is now, right? Now we've got jobs that range all over the place. So we have the jobs that range from science. And I have to say that one of the reasons, just to come back to why St. Louis too, is that we actually have some incredible scientists that had been working with gene editing for a long time, and we've actually developed a large portfolio now of very specific IP that's foundational to CRISPR. And what is that? That has allowed us to actually access hundreds of companies around the world that are working on CRISPR to talk to them about what they're doing. But that means that it's not just scientists who are developing CRISPR. So if you're a lab scientist and you're working with CRISPR, it's great. There's tons of jobs. We have about 80 scientists in St. Louis itself. So, but then you need patent lawyers because this field is really murky as far as the patent landscape. And if you want to develop something new as a startup, you need patent lawyers. So you need experts, legal experts in that area. You need bioinformaticists. Everything that we've been talking about involves the genome, 3 billion base pairs, you need ways of being able to access. If you start talking about patients, you need to be able to access that data and make sense of it. You need new areas that are coming up related to the bioinformaticists and computational biologists is AI, machine learning, better ways of being able to access this data so that now we can start to apply this as personalized medicine where we're treating every person individually rather than as a disease that affects 10% of the population. We're treating everybody as a single person. We need more and more in that area of being able to make sense of this data. And then after that, you need the people who are actually working with the patients, people who are able to take this research from the scientists all the way to the bedside. I'm sure I've missed groups.
How about bioethicists? You want to talk about?
Bioethicists are a huge part of it. And I will say, Merck, which we actually celebrated our 350th anniversary this year, so it's a whole different story for me from a startup. It's so young. But took our whole gene editing and our drive very seriously. And we have, and I think we're one of the unique tool supplier, vendor supplier companies that has this. We have a MBAP, which is a Merck Bioadvisory Panel, which is made up of ethicists from around the world, as well as legal experts and local, everything from religious to government officials in this panel. And what's very interesting for us is we actually watch them debate. We are not allowed to say anything. We bring them all together. We put up what our issue is, saying, for us, what will we sell CRISPR for? Because at some level, we're going to have to take our own responsibility. What will we sell CRISPR for? And then they debate it. We are quiet, which is really hard for a lot of us, because we all have our opinions. And we listen to what they say, and then we work with them to develop a principle. So we actually have a set of principles that all of Merck is trained on and we all abide by is that what we will and will not do in terms of selling our products to researchers or companies for use. So bioethicist is huge as well.
About genetic counselors?
Genetic counselors. WashU is just, I've been meeting a few of them and they've just increased their, I think their genetic counselors, but I think with things like 23andMe where people are starting to take the genetics, I think Dr. Dewan talked about it, and it's, as you're starting to take all that genetics, you want to make sure that you're as informed as possible about it. And that actually leads back to the education one, and there's a whole area of education because, and I think you were speaking about that earlier too, is that educating people to understand why changing something in a precise manner is very similar or is no different from years and years of breeding to get to that point. Why would there be, people are afraid of what they don't know, so education, and we do, and I think more, Pfizer does it, there's just labs where you can go in and work with CRISPR so that you get a comfortable feel for it.
Great. So the whole transformation from, we think we have a healthcare system today, but we actually have a sick care system. And what's going to happen with an example she provided is you actually are going to become a real healthcare system. And so that's going to create a whole set of new skill sets and jobs and roles and responsibilities. So that is great for on the human health side. Chris, anything you want to add to what Supriya said on that? And then I want to have Sam talk about how it's going to have an impact on the food and ag side.
Yes, yeah, sure. So I'll just kind of echo, I think what's important for different locations in St. Louis specifically is you think about building companies, growing companies. I mean, you do need a lot of these different skill sets here or able to import them in. And I think what we see in the, again, I'll go back to the Bay Area, in Kendall, Cambridge, is, data scientists, machine learning specialists that care about biology or care about agriculture and want to make an impact. And I won't throw Google or something under the bus, but so that's really important. And I think we have that here. So I think that's really important. And also too, we talked a little bit on the prep call about manufacturing. These personalized medicines, the whole manufacturing paradigm is continuing to change. I mean, we're investors in companies where they've built their own manufacturing. They're able to do it in a scalable way, but 2 years ago and what they're doing today are almost like night and day, right? And so you kind of have precision manufacturing for these products that are cheaper. You used to build a $100 million fermentation plant. Now you can get like $1 million clean room and put, some tissue culture in there to make these products, right? So that changes things. So do you have decentralized manufacturing? So you see a lot of this decentralization that disrupts the supply chain. And I think I can say one of the good things that Massachusetts has done a really good job of is 5, 6, 7 years ago the Life Sciences Center and Mass Bio started to invest in manufacturing education, and they kind of said, the R&D core is probably going to be in Cambridge because that's where everyone wants to be, but we can still create economically beneficial jobs to the broader market in cheaper kind of locations with manufacturing, and that's actually played out extremely well. Even though it's still more expensive to hire people and real estate's more expensive and all that stuff, people then want to be closer to their R&D hubs. So you start to think of these hub and spoke models from an employment standpoint. 10 years to play out, you have to be very strategic, but.
Great, Chris. Excellent addition to what Supriya had to offer. Sam, can you take us through, your vision perspective of how the whole ag food economy is going to get transformed, the kind of jobs that are going to get created or intensify, and the kind of jobs that are not going to be as relevant, if you will, in the new economy?
Yeah, I think that, first of all, I do believe that technologies like CRISPR and the newer technologies are going to mean that there are going to be more and more entrants into this startup world that address these food and ag problems, because that wasn't the case. This is a new thing, and it's just, I think we're at the very beginning of it, and this technology, CRISPR and the CRISPR-like technologies are an amazing sort of engine to throw that forward, and that's really exciting. And we see it. We see it here in this center. We see it on our campus. We see it on at the Ag Showcase. And Chris talked about the vital components. Another thing I think that CRISPR does is, when at the top of that list that Chris gave, is talent And one of the things that St. Louis has had a hard time is attracting, especially the 25 to 30-year-old, the millennials, the programmers, and with some of the stuff that Natalie and her panel talked about, sustainability, making a healthier, more sustainable food system, agriculture system. The kids, my oldest he'll be 24 in December. He's living and working in Boston. And his cohort, his age, really focus a lot on purpose-driven careers. It's not just a paycheck every 2 weeks and healthcare. It's, am I doing good in the world? And I think a tool like this really opens up the opportunity for folks to say, yeah, I want to be in the ag and food space. And they never thought of it before. And as we do a better job articulating the opportunities, and as we said, the grand challenge is in the next 35, 40 years, we're going to have to produce a heck of a lot more than the last 10,000 years put together, and you've heard it over and over again, using less things like fresh water and topsoil and all those other things. So that's really exciting, and I think it will turn heads, and we see it is turning heads. We're bringing folks who would have never thought, who would have stayed in the Bay Area or at Kendall Square saying, wait, I can really make a positive impact in the world. I'm a bioinformatician, and now I see I'm working with a research team, and one of the lead tools is gene editing, and we're going to do X and Y and Z, and that's going to be more food for people in the developing world and sub-Saharan Africa, using all that story. You can tell that story, and I'm excited about that. So those are some of the big things. I am, I don't want to be irrationally exuberant. St. Louis has got a ways to go. We've created a foundation. We've focused on what we're good at here, and we can make a case that show a diagram where we're best in class in these categories. I think we have to continue to double and triple down. We have to say to ourselves, how do we produce the next Benson Hill, and how do we keep them here? How do we keep them productive? And so it's incumbent on community leaders and others to do that. And believe me, I know that I and Dr. Carrington and others, we focus on this a lot every day. So,
great, Sam. Nice point you brought, which is about how to attract these young kids who are looking for purpose-driven careers and work. And it is inherent in the work here that it will be purpose-driven. Because of the nature of life sciences, whether it's on the human health side or on the food side. One of the other things that I wanted to ask you all about is the idea of being more cross-functional and multidisciplinary in how you approach this. You just laid out some of these things. Is that something that you think is going to be trending towards a greater intensification of a cross-disciplinary team more than before, and how that affects the kind of people that are going to be successful in this?
Yeah, there's a lot of the buzzwords, but I think it's more true than ever, given that there's all these more functional areas than ever, and they're deeper than ever, and they're more integrated than ever. And again, we go back to data science, machine learning, and biology and genomics, right? I mean, that you're generating so much data, and then you're layering on a lot of phenotypic data that's out in the public domain. And then you're still dealing with biology, which, all we learn every year is we don't understand biology. And then you try to bring in people with machine learning and bioinformatics skills to try to extract knowledge from this. We're actually able to do that a little bit now. There's probably a long way to go. But you can't really do that effectively if you're not in the same location. So we know a lot of our tenant companies, this is why we've been so successful, is because of this trend, or one of the reasons. If these people aren't together, you can't use Slack or Jabber or text message or Skype or anything. You kind of have to be in the same room and working on something and huddled around a computer. Nothing else seems to work as well as that. And when we're kind of at these bleeding edge areas where there's no paradigm, there's not even a common language to communicate, it's really the only way to do it. So it just becomes more and more and more important as you try to solve these harder and harder problems that bring all these disciplines together.
That's great. Anything you want to add, Chris?
I just wanted to add that, a lot of great science or a lot of great inventions happen when you bring two areas together that weren't there before. And, one of the ones that we're finding is engineers. So I think biology and engineers working together now is building so many more tools, because we're realizing it's not about the cell, it's about the cell in that environment, and it's about the structure of the cell. It's about whether it can contract or not contract, whether the pressures on it that keep it as an organ. So organ on a chip, where we can start to test different drugs and see how they're actually taken up by your organ, not by your cells. So we're trying to move to more system-wide approaches. That's where engineers and their understanding of torque and pressure and O2 concentration and how it affects the composition and structures, those things are all really coming out now because we're really bringing a lot of engineers into the mix of biology.
It's great. So question from the audience, what can be done on the state level to further develop the industry in Missouri? Sam, do you have any thoughts on that? And then, Chris, I'd love to get your point of view with what's going on in Boston that you may say, guys, you need to pay attention to, or you need to do more of. Love to get your input.
Yeah, I think there are a whole host of things from macro. We've got to do a better job educating our young men and women here, starting when they're in preschool, probably. We've got to do a better job making sure that our educational opportunities are put before all of our citizens, whether you live in a rundown community in the north side or in a distressed rural community. And so, when I was down in New Madrid talking to some of the parents, they don't have broadband. So their students don't have an ability to go online and take courses and do the Khan Institute and all the things that my kids who go to school in Clayton. So investments in tech and education and the infrastructure needed for 21st century educational opportunities. We have to make sure that our regulators understand the importance of tools like gene editing so that they make sure that our researchers can use every available tool to advance the cause of human health. We have to make sure that there are entities that help jumpstart the innovation ecosystems, things like the Missouri Technology Corporation, which takes an early, risky bet on the very early stage companies where they're just not ready, they don't have a balance sheet to go to traditional sources of capital. Critical. And I think that we all have to do a better job getting out and talking about the importance of what we do. So I think there's a lot of opportunity there across the board.
Yeah, as Sam was talking about it, a thought that ran through my mind was, if we did a survey of the residents of Missouri on their thoughts about gene editing, the attitude towards it, how will it index versus some of the other regions of the United States? And that ought to be an interesting starting point for what's the gap to close or what's the advantage to build on. Don't have an answer to that question as far as I know. Anybody in the audience has it, love to share it with the folks here.
Well, with the folks I was just with, if I said, what do you think of gene editing? They'd say, I wish they weren't so tight. So it's...
There you go, Sam.
That's the other kind of gene.
That's the other kind of gene. All right, Chris, you're going to have to follow on that one. So tell us about what's going on in Boston that you think would be really great for us to, pay attention to and benefit and leverage.
Yeah, so I think I'll speak probably a little bit high level because I'm not as familiar with the St. Louis and Missouri market as I am with others, but I do know from being here there's a lot of really great groups. We emailed about BioGenerator. I've gotten to know those guys. They have someone based in Cambridge that I know pretty well. So there's kind of this early stage public-private partnership, risk capital. There's kind of regional economic development, focus economic development, technology economic development. I think what I've seen, and I won't call out from the other markets I've seen, but I think one of the things Massachusetts has done really well with the Life Science Center is say, here's the first initiative was a 10-year, $1 billion budget, $100 million a year roughly, and they just did that. So they gave them long-term, so government should be long-term planning. I can contrast with other regions we're in where these organizations are a line item on a budget. So how do you retain good people? How do you predict? How do you invest? So you need long-term planning. You need a commitment. And then provide that group with flexibility to kind of invest where there's need, right? And either leverage that with private capital or with the Danforth Center and try to identify unique opportunities within the marketplace that will be a long-term investment for the future. So I gave the Massachusetts example of biomanufacturing, right? I don't think any of us foresaw the CAR-T manufacturing, the gene therapy. You can't get AAV manufactured for 12 months right now. There's such a supply and manufacturing crunch. I don't think Massachusetts foresaw that. Maybe they did. I wish they would have told me. But things like that. So think about what's 5, 10 years out. What can we invest in today that will make us a magnet for the future? And also think about what do you have here today and capitalize it when you can. So a good example, again, not that the wasn't necessarily an involvement, but if you look at we have a big presence in Maryland. In the genomics era, that region was gangbusters. The Human Genome Institute, a bunch of great organizations. And then when the genomics era didn't quite pan out, everyone thought it would. We didn't cure disease overnight. It slowed down, and Cambridge and San Francisco kind of took over again. What we've seen now is with, again, immuno-oncology, infectious disease, immuno-oncology, and immune system. It's a very strong infectious disease vaccine marketplace. People can't find immunology talent, and people that understand vaccines understand the immune system, turns out. So that market has been a big draw for this kind of new wave of technology that's finding an old skill base of talent where they couldn't otherwise find it. So I'd say kind of just really broadly assess what you have here from a core underlying, and I know it's very diverse and very broad and really strong in some areas, but broadly, what do you have as a talent base? And what can you do today? What can you invest in that you see coming down the pipe? And then position it so when things that are unpredictable do come, which everything in biology seems to be, you're ready for that. And I think this gene editing thing is a great example of that. I mean, there is a lot of great talent here, both on the agriculture and healthcare side addressing that. And I'd say, it's a good opportunity to double down and leverage that. But think about what's gene editing 3.0, 4.0, 5.0 look like. Because it's here. That's here to stay for sure. So, but I would say longevity and flexibility, and that'll give you good people, and then good things will happen.
Great, Chris. Supriya, anything you want to add on what can we be doing here that hasn't been covered?
I think you guys covered it really well. I would add, though, I think meetings like this one, where we're bringing together a lot of different people from different groups together to really talk about, I think we talked so much about the promise. I think you could have probably the same one about the obstacles and the challenges that we have there as well. And I think bringing everyone together to do that like this is key. It was really good for me listening to the other one.
Yeah,
Discussions this morning.
I agree again, like that informal network of people that have worked together and then now do different jobs, and then their networks just kind of expand. Like that's how business deals, transactions. You go, probably know the Broad Institute, right? When I go there for a meeting, I sit in the lobby, I see some scientists come in, hand them a tube, and then walk out. Probably no MTA there. But that science was just sped up because those people knew each other and trust each other, right? So it's a very magical element that meetings like this and a lot of other things make happen.
Yep. Yeah, Sam, what may not be well known to folks around here is how much of that actually is going on here in the ag tech space with all the startups. Can you paint a picture of how the Danforth Center sits as the hub of a beehive of innovation, where there's a Benson Hill and there's an Arvigenics and there's the Evergenes and these companies from around the world that are coming to St. Louis, just to help to crystallize Chris's point here.
Yeah. So we sit here physically in the center of the 39 North District. Which is important. I mean, there's just geographic. You can walk.
I'll interrupt you. How many of you know what the 39 North is here? Oh my God, this is an incredible audience. You guys know what you're talking about. Okay.
So, and that's important that, there's physical proximity. Benson Hill at Helix is just a short, we're hoping to get a short walk, but it's close. And then the folks at Bridge Park are, 100 yards to the greenhouses and other cores, and here. But more importantly is that we're culturally connected, that when we understand that our mission is improve the human condition through plant science, that discovery by itself won't achieve mission impact, is when that discovery gets out into the world as a product or a service or collaboration with Sigma or working with the Benson Hill or helping to launch a new startup. And here at the center, we see folks from the Benson Hills and the other companies here every day, walking the halls, interacting with our investigators, eating lunch in our cafeteria, and vice versa. We're there, and so that's really critical, is the cultural piece. And I think you can have the best physical layout, but if you don't have the right cultural piece, it won't work. And so I think that's a real area of advantage for us, because I think we're really on the vanguard of this sense of, we have a number of core facilities here, 6 core facilities from growth rooms and chambers and greenhouses and and we make all of them open to the community. They pay, but it's just in time and is as needed. And that openness and connection helps build relationships between our core facility managers and our investigators and the startup world. So it's really critical.
Great, Sam. So what you all may not know is that we have a vibrant network of international ag tech companies that are all coming into St. Louis after looking at all the other locations in the US where they could go, and we usually are not the first place on their list. We end up becoming the place that they come to. And one of the reasons is what Sam was talking about. You can create a picture like this if you go to RTP. You might create a slightly different picture when you go to Davis in California. The thing that makes a difference is that this picture in St. Louis, we are actually working across and working with folks in these different bubbles in a seamless way. And that's what these folks experience. There was a senior leader from Bayer who told me that, Vijay, now that I've been to St. Louis a few times, we've got a similar picture in RTP, but in St. Louis, it really, really works. So that gives you an example of the cultural aspect being so important. And this is what you see in these really innovative ecosystems like Boston and San Francisco. And we have built it in our own way, but we have that. So my calling to all of you, as you are here today, is really get to meet as many people around the room, get to know each other, and you'll be surprised how there is a great cross-linking between the work you're doing and how somebody across the room could actually advance it or could be a partner with you. And if we keep doing this as a community, cross-linking with each other, we will keep separating from our competition. So with that, let's end this topic about the 21st century economy. Go cross-link. Take care.
I just wanted to thank all of our speakers again for taking time out of their busy schedules. Thank you so much. I also would like to thank the Danforth Center and Benson Hill for being our co-host, and then Dr. Carrington for hosting us here at your beautiful facility. And also thank my sponsors, and finally thank all of you for taking the time to come and be a part of this symposium. Thank you.